The Australian Institute of Physics, the Teachers Guild of New South Wales, the Royal Society of New South Wales and the Royal Australian Chemical Institute acknowledges the traditional custodians of the land on which we meet here this evening at the Concord Golf Club, the Wanggal people, and pay my respects to the elders past, present and emerging. Ladies and gentlemen, my name is Dr. Frederick Osman. As the President of the Teachers Guild of New South Wales and the Secretary of the New South Wales Australian Institute of Physics, I'm pleased to welcome you this evening at the Concord Golf Club for our 2024 Frontiers of Science Forum presented by the Australian Institute of Physics, the Teachers Guild of New South Wales, the Royal Society of New South Wales and the Royal Australian Chemical Institute, in support with Laboratories Credit Union. Tonight, we are very privileged to join by Mr. Mark Currie, MP, the Member for Oatley, Shadow Minister for Multiculturalism, Shadow Minister for Jobs, Industry, Innovation, Science and Technology, and Shadow Minister for the South Western Sydney, along with Professor John Maddock, Dr. Renee Gorman, Professor Katrina Jolliffe, and Associate Professor Richard Garner as our speakers at this year's combined Frontiers of Science Forum. I would also like to thank Mr. Ian Wolfe from Diffusion Radio, who will be involved with our panel discussion later this evening on exploring major discoveries and theories of physics, mathematics, biology and chemistry. The format schedule of this evening will be our presenters to speak for 25 minutes, each on their chosen Frontiers of Science topics. And this will be concluded by a panel discussion and a Q&A at the end. So please hold those questions to the panel sessions. We are also videoing this event so we can make it available for everyone that could not attend tonight, especially our regional areas. It gives me great pleasure to introduce Mr. Mark Currie, MP, the Member for Oatley, Shadow Minister for Multiculturalism, Shadow Minister for Jobs, Industry, Innovation, Science, Technology, and Shadow Minister for South Western Sydney for being here tonight. Mr. Mark Currie has been a member for Oatley since 2011. Mark lives in Penthurst with his wife, Adler, as a school teacher, and their two sons, James and Sam. Mark now has the honour of serving the Shadow Minister for Multiculturalism, Shadow Minister for Jobs, Industry, Innovation, Science, Technology and the Shadow Minister for South Western Sydney, under the leadership of the New South Wales Opposition Leader, the Honourable Mark Speakman, SC MP. With his current portfolios, his passion in supporting promoting multiculturalism, he's got a great support for science technology, and we are very privileged for him to be here today. Ladies and gentlemen, please welcome Mr. Mark Currie.
Well, ladies and gentlemen, it's a great honour to be here today, and I want to begin by acknowledging the traditional owners of the lands in which we meet today and pay my respects to elders past, present and emerging. Yes, Frederick, you're absolutely right. I did marry a teacher and I married up in the world. And a wonderful teacher she is. And I do have two young boys, as I call them, Hurricane and Tsunami. But they've got a love for science. And I remember as I was driving in, I was reflecting my passion and love for science began at a very young age. And I remember one of my very first chemical it was a chemistry test kit and Bunsen burner. And after almost burning the house down when the Bunsen burner fell on the tablecloth, it was just a chemical test kit. But I remember that passion for science, and it's something that I have passed on to that next generation, to my kids, particularly James, who is celebrating his 10th birthday tonight. But don't worry, I'll be there in a sec to celebrate his birthday. But it's that passion for science that we've got to, follow through with as teachers, as parents, as, that generation, so that the next generation can continue that passion. So it is wonderful to be here today, this evening. Innovation, science, technology is the engine room that drives positive change. Change in industries and transforms the way in which we live. And they are the driving force behind economic growth and prosperity. And in the 21st century, it's something that government needs to be backing. And during our time in government, or my time in government, we had a state election last year. We had a strong record of supporting the innovation, the science and technology in our state, in our great state. And so much so, New South Wales saw over a 300% increase in innovators between 2013 and 2022. We introduced an R&D roadmap setting out a 20-year vision for New South Wales to produce more world-leading new technologies, products and services. And we backed it in with funding. We saw over $700 million, a future economic fund, delivering the state's first fund aimed at a full life cycle of innovation, of high growth business in a number of different priority sectors, and drive productivity at the same time in emerging high value industries such as digital technology, such as medical technology, such as the clean economy, of course. And we also invested over $270 million in biomedical technology, attracting world-leading researchers, scientists, clinicians to our medical precincts, and delivering those breakthroughs that create a brighter future for people everywhere, not just in New South Wales, but around the globe. And under the current government, we hope that these investments will continue, particularly as industries across innovation, science and technology face many different challenges. And as you know, major discoveries, theories in physics and mathematics, biology, chemistry, have shaped and continue to grow at an enormous rate. Because this financial investment here in this industry, or industries I should say, needs to be targeted. So grant programs cannot afford to take 6 to 8 months to distribute funds to researchers, and delays can be critical in this space. Additionally, I have to say, department and bureaucratic structures and hierarchies hinder the flow of innovation thinking and slow down that implementation of groundbreaking concepts. This is why forums like tonight are so important. Collaboration is such an important key. Hearing from a range of experts, international experts here tonight, on the latest and the future development in their fields offers insights as well as inspiration for the kind of research that can and is certainly being undertaken here in New South Wales. And it also shows where we're heading in the future of mathematics, in physics, in chemistry and biology as well. I must say New South Wales has been and needs to remain that driving force of innovation, science and technology. Because when New South Wales is at the forefront of innovation, science and technology, Australia becomes a leading nation globally. And certainly as the Shadow Minister for Industry, Innovation, Science and Technology, and I hope one day to be Minister again, I've heard from a range of key stakeholders over the last 12 months on what are some of the important issues affecting them, affecting their research, and affecting these key industries. So there are 5 themes that many issues have been brought to my attention appear to fall under, and these have since become 5 key areas that I'm quite passionate about, in which, as a shadow minister, I've been advocating whilst we're in opposition, advocating for the government to implement. And some of these are, for example, 1, continued investment in innovation, continued investment in science, continued investment in technology. And whilst government regularly provides funding to help startups and companies establish themselves in New South Wales, there now needs to be sustained support to ensure that the work being undertaken by these companies here in New South Wales can continue to grow and to expand here in New South Wales. And this doesn't only mean, I should say, making additional financial investments, while it helps. It also includes how we can introduce or amend government policy across government to make it easier for research and development to take place. All types of research and all types of development to take place. The second point is focusing on those new and emerging industries here in New South Wales. Last year I began hearing that the key space and defence stakeholders are now starting to look away from cementing the space and defence industries in the Western Sydney or around the Western Sydney aerotropolis. And this is an example of what happens when different levels of government don't focus on those emerging markets or those emerging industries. Instead, we need to be supporting them at greater knots. Building up our emerging industries is more than just about economic benefits. They are building an ecosystem of jobs for the future of young kids, my hurricane and tsunami. New South Wales can't just be a service economy. And to be an economic powerhouse for the future, for your kids, your grandchildren, for my kids, New South Wales needs a strong base grounded in innovation, grounded in science and those technology sectors. The third point that I've come across is, of course, building a pipeline of apprenticeships and trades. And as an emerging issue that I've been hearing from stakeholders in a number of different sectors over the months, particularly for innovators, is that there is a difficulty in recruiting people into jobs across the innovation, across the science and technology sectors. And to deliver this or to combat this, government needs to work closely with stakeholders and investigate how we can combine micro-skilling with the current and future courses offered to ensure the future workforce into the future is well equipped across all levels of government. The fourth, of course, is investing in tech and startup precincts. And this is exciting stuff, and I have visited a number of them over the last few years. Collaboration is really the key. And when it comes to tech and startup precincts across New South Wales, or across Sydney at least, but definitely across New South Wales, that collaboration is so important. And even with technology allowing people to connect remotely, in-person research, collaboration is still what drives the advancements in our key industries. So government needs to be looking at ways, new and improved ways, and how we can continue to support precincts like Macquarie Park, the future Tech Central, the Western Sydney Aerotropolis, which will be a complete game changer for not just Sydney, but for New South Wales and, of course, across Australia, to ensure that places for innovators to collaborate are created. And, of course, the fifth is supporting STEM in the education system. This connects with point 3 regarding the pipeline for the training, the pipeline for the apprenticeships. And I'm sure this will be spoken about by many of you here tonight. In order for us to encourage the next generation to be part of the workforce in innovation, science and technology fields, it is essential that our kids build an interest in science and technology. And this, ladies and gentlemen, starts at school. So, unfortunately, whilst the former government was seeking to make mathematics compulsory in 11 and 12, perhaps even science, the current government have announced recently that they will not be pursuing this. And I have to say personally, this is quite disappointing. I will continue to be advocating for mathematics at the very minimum, but also some sciences as well, to be compulsory across 11 and 12. Students need the academic foundations and the critical thinking to thrive in that competitive, globalised workforce. Whether it's for everyday problem solving or preparing for the future jobs in space, technology, science, engineering, STEM is so important. It is so valuable, for students, and governments need to better recognise and support this. Our state cannot attract the investment, the the support industry, and increase and protect local jobs without investment in engineering, without investment in science and technology education. And I hope all of you are looking forward to the presentations that you hear tonight. This research demonstrates the breadth of where career across science and technology can take someone, and the benefits this research will bring to improve people's lives and creating a better future for everyone in our great state and country. As educators, whether at a high school or a university level, the work you do to foster a culture of interest in technology is truly important. And I'd like to thank all of you for the work that you do across the many fields in this space. Finally, thank you again for having me tonight. As I said before, it's not just an interest, it's a passion of mine. And I hope to meet many of you into the future and to the many other science forums and science expos that pop up from time to time, and certainly to hear your thoughts and suggestions on what we can do to progress the areas of science, technology and innovation into the future. Thank you very much.
Thank you, Mark, for your support, for being here tonight and setting a wonderful platform for us for our presentations. Please accept a small token of our appreciation as a lasting memory of this event from the Combined Associations. And we appreciate you being here. Enjoy it. Thank you. Our first presenter tonight, John Mattick, is a Professor of RNA Biology at University of New South Wales, Sydney. He was previously a Foundation Director of the Institute of Molecular Bioscience at the University of Queensland, Foundation Director of Australian Genome Research Facility, Director of Garvan Institute of Medical Research, and the Chief Executive of the Genomics England, where he developed the 2019 to 2023 UK National Vision and Plan for Genomic Healthcare. Professor Mattick is best known for showing most of the human genome is not junk, as previously thought, but rather is devoted to controlling human development. He has published over 300 scientific articles, which has been cited over 98,000 times. His honours and awards include Australian Government Centenary Medal, the International Union of Biochemistry and the Molecular Biology Medal, the University of Texas Bertner Award for Distinguished Contribution to Cancer Research, and the Human Genome Organisation Chen Medal for Distinguished Achievement in Human Genetics and Genomic Research. Ladies and gentlemen, please welcome Professor John Mattick.
So I'm here to tell you that we molecular biologists have fundamentally misunderstood the structure of genetic programming in humans and other complex organisms for the past 60 or 70 years because of the apparently reasonable but ultimately incorrect assumption that most genes encode proteins. There's a gene for hemoglobin, a gene for insulin, etc. And that was developed out of bacterial genetics, very simple organisms, and it was just simply assumed that you could extrapolate what people in the 50s and 60s and 70s discovered in microorganisms to humans. I want to start with one of my favourite scientists, Barbara McClintock, and if you don't know Barbara, you should. She's probably the giant figure of the 20th century in genetics, and this is her as a PhD student in front of her beloved Mays at the University of Missouri. Now, she got the Nobel Prize in the 1980s for discovering transposition, that is, genes that jump around, which she saw under her microscope change the phenotype of the corn. And she intuited that genes did more than encode proteins, the component parts. And so this is an excerpt from a letter she wrote to a good friend in 1950. Now, this is 3 years before the double helix. She says, "Are we letting a philosophy of the gene control our reasoning? What then is the philosophy? Is it valid? When one starts to question the reasoning behind the present notion of the gene, that is, as a protein coding unit, likely, which was held by most geneticists and the biochemists, the opportunity for questioning its reasoning its validity becomes apparent. What she was really saying was, and it was a deep intuition in the best sense of that word, that there are other forms of genes that change the patterns of expression of the proteins, and that she could see in studying her maze. Unfortunately, she was ignored. She got the Nobel Prize, but she went to her grave as the great dame of Gold Spring Harbor Laboratories in New York, frustrated that people didn't understand that it was about regulation, control. Although she was fated, she was elected president of the American Society of Genetics in 1948, so she was valued, but her intuitions were not appreciated. So as I said, the assumption, based on the studies of what's called the lactose operon in Escherichia coli, one of our gut bacteria is very heavily studied, has been that genes are generally synonymous with proteins, that most genetic information, including regulatory information, is transacted by these proteins. And this protein-centric view of the programming of life reflected the mechanical zeitgeist of the age. Now, there's only a few of us in the room old enough to remember, but back in the 50s and 60s, the world was made of bicycles and smokestacks. Nobody thought about digital programming or any of the more sophisticated views of information. Anyway, it led to many subsidiary assumptions, despite a number of surprises that should have given pause for thought. And this is part of the history of science. You get surprising, shocking observations that people just sweep under the intellectual carpet because it doesn't fit their view of the universe. So the first surprise, which is where I got interested in this, back in Texas when I was working there, is that genes in humans and other complex organisms are mosaics of protein coding and non-coding sequences. So there's a bit here, a few words, and then a stretch of junk, apparently, a few more words, so forth. And they were called intervening sequences or introns. And immediately in university, despite the fact that they are copied into RNA of the gene, DNA makes RNA, they were condemned as junk. But the equally, if not more plausible and far more interesting alternative is that other information is being transacted by these RNAs, which is what popped into my mind after 2 beers in a Texas pub on a Friday night. It stuck with me. Second surprise is that our genomes are full of retrotransposons, viruses, all sorts of things that were condemned as selfish DNA, but these turn out, I don't have time to give you examples, but the major drivers of evolutionary change. The third surprise was that the number of genes in complex organisms doesn't scale with developmental complexity. And this was rationalised in various ways, but let me just illustrate this. And I've got a comment here. These enduring interpretations are examples of what's called founder fallacies. Now, founder fallacies are when people make observations in science and they try naturally and properly to generalise from those observations. The generalisation, if it can't be tested immediately, becomes the orthodox interpretation. Then, sort of drifts into being an article of faith. And you've sort of got knocked down buildings before people will reassess the initial guess at what these things meant. So let's look at development. Now, there's 2 organisms here, both animals. The one on the right is a nematode worm. It's 1 millimetre long. There's about 1 million of them in every cubic metre of soil out there. They're most populous organisms on the planet, far more than insects, and they only have 1,000 cells. The organism on the left, that's us. It's got bones and muscles, and I'll illustrate this in more detail in a moment, and it has about 37 trillion cells, opposed to 1,000. But the genome projects, which came around at the turn of the century, once the DNA sequencing technologies had advanced sufficiently to do that, showed that they both had the same number of protein coding genes, about 20,000. Bloody hell. And it turns out even worse that most of these genes are orthologous. They fulfill the same functions. There's muscle proteins, nerve proteins, etc. And many are common with simple eukaryotes like baker's yeast. Now, the big shock that came along with this was that only 1% of our DNA encodes these 20,000 protein coding genes. So everybody assumed the rest must be junk. I mean, amazing. The question rises is how much information is required to program human development from the moment of conception to adult, and to program our cognitive capacity, and what form does it take? Now let's have a look at this. I love bones. Bones are the most fascinating organ in the body apart from the brain, because the architectural variety is extraordinary. So these are 3 vertebrae from different parts of the spine in 3 planes, and any anatomical pathologist can look at them and say that's C2 or L3. Look at the architecture. Every bone in your hand, the flat bones in your face, your pelvis, long bones in your leg, all have to be programmed to precision from the point of conception. Look at the muscles. Every muscle's different. Every muscle's got a different connection point, ligaments. Plus you've got all the organs and of course the brain. So in C. elegans, that nematode worm, 1,000 cells, the complete history of every cell in that worm has been mapped under the microscope from the point of conception. The same precision has to occur in humans. If you look at identical twins, they're phenocopies. So the differences between us as individuals is not, in utero problems or variations, it's actually variations in our sequence. So in the case of the worm, when you've got 1,000 cells in that tiny little thing, it takes 2,000 cell fate decisions. In our case, we have something of the order of 80 trillion cell fate decisions have to be made with perfect accuracy from the moment of conception. And so I think you'll see where I'm going. There's only a small proportion of our DNA is actually devoted to the component parts, the muscle proteins, or hemoglobin, and to our biochemistry and physiology. Nearly all of it is devoted to our development. So the first clue broadly to this was that the amount of non-protein coding DNA increases with developmental complexity. As a proportion of the genome, the genomes get bigger. And we now know that irrespective of the extent of these so-called non-coding sequences, they're all transcribed into RNA dynamically in different cells and tissues at different developmental stages. So you're forced to the conclusion either there's an enormous amount of useless transcription or there's another layer of information that's being transacted by these RNAs. Now the data supporting this came out of studies on high throughput RNA sequencing initiated in the early 2000s. And we were part of this group in Yokohama. And these studies were motivated by trying to use high throughput RNA sequencing to define the different proteins and their isoforms. But the unexpected and ultimately headline result was that there were tens of thousands of RNAs that didn't code for protein that were being produced. Excuse me. And we showed that many of them had overlapping antisense transcripts from the other strand and had some evidence that these were regulatory. So what came out of this, and this is 20 years old now, is a view of the mammalian transcriptome. That's all of the RNAs that are transcribed from our genome as being a continuum of coding and non-coding transcripts that overlap. Their splicing patterns, their promoters, their finishing points, etc., vary in every tissue and cell. They're stable, just like protein coding RNAs, and they have all of the indices of being real genes. I won't go through the list, it's a bit technical, but I just go to the last 2 things. They show very specific expression patterns, much more specific than most protein coding genes. So here's a study we did with the Allen Brain Institute in Seattle many years ago, looking at the expression of some of these RNAs in the brain, mouse brain. And the primary data is in the top set of panels, then computer transformation, second panels, which gives you better contrast. So if you look at panel B here, this one, there's an RNA which, as far as we can tell, is only expressed in the core of the memory centre in the hippocampus, called the dentate gyrus. This one's only expressed in the CA1 subfield, and so forth. Extremely precise, much more precise than protein coding genes, which tend to be more generic parts, as I said. We found lots of these things. We also found that they were trafficked to very specific places in the cell, to the cytoplasm, mostly go to the nucleus, some decorating unknown structures still unknown that we're trying to purify, others that we have characterised, and then some go into the ends of nerves. And one of these things, just to give you an illustration, was first discovered by a Japanese group called Gamafu. We were able to show that this thing is acutely downregulated in response to neuronal activation and has all of the hallmarks of the genetic changes that are associated with schizophrenia, and is one of the prime candidates for the variations that cause that disorder. Now, it gets a bit technical, I'm sorry, but try and skim through this. But large studies of genomic variation around the world with tens, if not hundreds of thousands of people trying to map the genetic variations that affect complex diseases and disorders and traits have shown that they map mainly to non-coding regions. And they generate this sort of plots, and here's the one for intelligence. So there's a whole genome, and then there's the signal in different parts of the genome, and they're called Manhattan plots for obvious reasons. So where the dots go right up, that's a place in the genome that's associated, in this case, with intelligence, however that was measured. So it turns out that hardly any of them are in protein coding regions. They're nearly all intronic or intergenic, that is non-coding, which means they're regulatory. And it's well accepted out there in evolutionary biology that the differences between species, what's called adaptive radiation, whether it's in the primates or the mammals or the vertebrates, is mainly due to regulatory variation changing the patterns of gene expression rather than the nature of the key components, although they do evolve slowly as well. So some years ago, and Kate knows Marcel, who's now Dean of Science at the University of Sydney, and his great student Ninat, and the rest of us looked at the RNAs coming out of regions associated with all these different sorts of complex traits and disorders. And here's one example. The red bar there is the region of the genome that's associated with cognitive performance. Nothing in the database down the bottom. This is the tag we use, and you can see that right in the middle of this locus associated with cognitive performance is a multi-exonic, long non-coding RNA that doesn't code for protein, which really has to be a prime candidate for whatever the mechanistic basis of superior cognitive performance may be. And we in fact showed most of these loci identified by these genetic studies express these RNAs. And we also showed that they come out, don't bother reading the text, they're associated with those involved in neuropsychiatric functions. We also showed, as did other groups, that these RNAs are very dynamically expressed during differentiation development, embryonic stem cell development, neuronal stem cell development, immune cell activation, muscle differentiation, breast cancer and mammary development, etc. And over the years, the evidence has been slowly accumulating that these RNAs are involved in all sorts of developmental processes, and this is just a sort of list: brain development, limb development, etc. And here's one example of a thing which is called CodeChaser, which when is in heterozygous state produces skeletal abnormalities because the mice don't develop properly. They're also involved in cell fate programming, reprogramming, and are central to stem cell biology, and will be major players in the commercialisation or use of these technologies to improve human health. Marcel and my group and another colleague, Paulo, who's now in Brazil, showed that these RNAs are very dynamically expressed during embryonal stem cell development in vitro, in culture, and showed that, and I won't bore you with the details, but showed for the first time that these were associated with chromatin modifying complexes and modified histones. Now the DNA, I should say our friend John Rinn also showed a year later that most of these RNAs are actually associated with chromatin modifying complexes. What are they, you say? Well, they're the basis of epigenetic processes. So epigenetics means the chemical modification of the DNA, and mainly the proteins involved in the histones, which are the core structural unit of our chromatin. So our DNA is not just a string, it's wrapped around these proteins called histones in structures called nucleosomes. And these things are essential for development, long-term responses to environmental pressures, including things like type 2 diabetes and cancer, and also essential for brain function. If you knock out epigenetic processes, you cannot learn. Now, these chemical modifications are catalysed by a suite of enzymes that have no sequence specificity, but impose these chemical modifications, hundreds of them, including covalent linkage of neurotransmitters in your brain, all sorts of really interesting chemical modifications that feed into epigenetic memory and epigenetic control of development. And they occur at hundreds of thousands, millions of different locations around the genome, different cells at different stages of differentiation. So the question arises, what determines the site selectivity of these enzymes? What determines the positioning of these structures? And what's the molecular basis of environment epigenome interactions? And the answer to all three of these questions is RNA. So it turns out that these RNAs are produced from genetic loci called enhancers, and most people in the room have never heard of these. And they only appeared as a term in the 1980s, although genetic evidence for these things occurred much earlier last century. So, excuse the text, but I've just got to whip you through this. Turns out most of these RNAs, these tens of thousands, hundreds of thousands of RNAs, are actually the products of genes called enhancers that don't code for proteins. So enhancers are genetic loci that control the spatio-temporal patterns of development, and that's been known for a while. If you muck around with them, you mess up development. They're estimated to be of the order of 1 million, give or take 500,000, so a rough estimate, in the human genome. These things, like protein-coding genes, express long, multi-exonic spliced RNAs in the cells in which they're active. Knockdown studies genetically have shown that these RNAs are required for their action of these genetic loci that control the spatio-temporal patterns of our development. And the production of long coding RNAs from these things called enhancers explains their cell-specific expression, like I showed you in the brain, their rapid evolution under positive selection for adaptive radiation. So the difference between us and other mammals is not the protein coding set, it's actually the patterns of expression, longer teeth or limbs or whatever. And the genome-wide incidence of all of the signatures of active genes. This also resolves the so-called G-value enigma, which is, the G-value enigma was 20,000 protein-coding genes in a tiny worm and 20,000 in us. That's an enigma. In fact, we have around about 1 million genes, whereas the worm doesn't. 20,000 of which can code for proteins, and the rest of which code for these RNAs that organise the patterns of expression. So they're the architectural plans. And this is, now it's getting a little technical, but I'm almost done. This is probably the most exciting area of cell biology that's emerged in the last decade or 15 years. And that is our cells are actually very beautifully organised internally, not just with membrane-bound structures like mitochondria, but microdomains that are actually liquid physics, phase-separated domains that organise virtually the entire nucleus and the cytoplasm. Even translation is a phase-separated domain. And here's some pictures of long non-coding RNAs, and you can see the dots. They're in very precise locations. And probably a bit technical, but basically these RNAs are forming, binding to the chromatin to bring it into a complex that then determines which genes are expressed in the next phase of cell division. Because remember, from the moment of our conception, 80 trillion decisions have to be made with accuracy. Divide or differentiate, divide or differentiate, divide or differentiate, all the way up to birth and puberty. And if you stain for RNA talking to DNA, in other words, RNA-DNA structures, the nucleus looks like the Milky Way. It's all over the place. Now these RNAs are actually scaffolds that bind proteins that can modify the chromatin, epigenetic marks, transcription factors that determine which genes are transcribed, and also then bind to other RNAs or DNA to target these complexes to their right places. So here's a view from an article I wrote last year. So in 1980, the amount in blue there at the top about how much was protein coding was not known. It was speculated 100,000 genes, 500,000, but once the genomes have been sequenced, it turned out only, in our case, 1% was protein coding, another 1% regulatory for those sequences, and the rest was thought to be non-functional. But today, we now know the whole genome is functional, and most of it is devoted to the production of these enhancer RNAs that control our development from conception. We also know, and I don't have time to talk about this in detail, but RNA is the substrate for the environmental interactions. So the RNA is extensively edited and modified in response to environmental signals, particularly in the brain. So what's hardwired to control our development, to put fingers and toes in the right places, is then softwired in the brain by superimposing chemical modifications. Now, there's fantastic commercial opportunities here because we can use our knowledge of this to manipulate immune responses, better vaccines, better cancer therapies with CAR-T therapy, if you've heard of that, and autoimmune disorders, which are probably the major source of the health burden. So we're actually working to start companies in that space. So to finish, you know, the idea that was popular when I was a postdoc, and just from lazy assumption that what came out of bacteria was extrapolatable to us, used to be that the human genome was kind of oases of protein coding sequences in a desert of junk. But in fact, the reality is that our genome is probably the most sophisticated information suite that we have yet encountered in nature, because the amount of cell decisions, those 80 trillion, is actually 10,000 times more than the information content in the genome digitally. So the real view of the genome is not oases of protein coding sequences in a desert of junk, but islands of protein coding sequences in a sea of regulation. And that's what puts us here today, and what makes us different from each other and other species. And I should say most of the work was done at the Garvan and also my old institute in Queensland, and I want to thank people who've contributed. Thank you very much.
Thank you, John. Always exciting to hear wonderful research in the area of RNA. From biology to physics. Dr. Renee Gorman is a senior lecturer in physics at the University of Newcastle and leading expert of nanobiotechnology, having earned her PhD in 2014 from the University of South Australia. She has since held notable postdoc positions at Flinders University, the University of South Australia and Victoria University of Wellington. In 2019, Dr. Gorman joined the University of Newcastle, where outstanding contributions were acknowledged with the Women in Research Fellowship in 2020. Her research, deeply rooted nanobiotechnology, allows for multidisciplinary research teams to form. In 2023, she was awarded the AIP New South Wales Community Outreach Award in Physics for her contributions to disseminating science to the community. Ladies and gentlemen, please welcome Dr. Renee Gorman.
Thank you, Fred, for inviting me, and AIP for also allowing me to present today. Suzanne, I will have a trifecta. I am a member of RACI and AIP, and I will be a member of the Royal Society of New South Wales tonight. Sorry about that, I'm one of those two. Yeah, I had a little giggle when Fred introduced me because I am a physicist. I'm in the physics discipline, but my research has some biology in it. So I'm not an expert though, so I have experts in my collaborative space. So the title here, Don't Hold Your Breath, please don't. I'm looking at making a disease breathalyzer, one for lung cancer, and more recently one for viral detection, but that's in its infancy, so I'm not talking about it today. I wanted to talk a bit about me because I did get the Outreach Award, and I wanted to discuss a little bit as to why I really love to do outreach. This is me. I was born in Karratha. I have a twin sister, so there was two of me. She's completely different. She's in the corporate role and earns way too much, more than I'd ever dream of. Yep. We moved around a lot in WA and South Australia, and that's where I did my PhD, which has already been said. So I did my PhD at University of South Australia, and that's where I fell in love with nanotechnology. It was after postdocing between the two universities, Flinders and UniSA, that I decided to drag my family. It's a funny story because my husband's English and he hates cold weather. So I had to drag him quite literally to Wellington. Luckily there's good surf there. I had a 5-year postdoc position, and that's where I shortly after started a lecturer role and then came back to my family. Oh, well, sort of, because they're on the other side. But within the same continent. So I've been at the University of Newcastle for 5 years, so it's my longest job so far. This is my research group. They're the ones who do the research. I just tell them ideas and come up with solutions sometimes. So I've got some past students. Garima basically incepted this project. She's the one that I worked with in Wellington and came up with this idea. And now Zarina, she's got an amazing story. She came from New Zealand to do a PhD with me, then got stranded back in New Zealand for COVID, and has done a complete PhD there. I didn't tell the university, keep that one quiet. But yeah, so she did an entire PhD at Christchurch at a surrogate lab, and that's what today's research is based on. So now we have Emma who's continuing that work. And obviously a lot of other great researchers there too. Like I said, it's a multidisciplinary team. So we look at E. coli detection or pathogen detection, lung cancer detection using similar platforms. I use quantum dots. I love quantum dots, which I'll discuss a little bit today. And also these guys here are Lee and Anish. They're looking at battery storage. So they're looking at microemulsions for electrolytes. Very broad. So lung cancer. We know that it's quite hard to detect, and usually people don't go to the doctor until they have symptoms. So that causes a late diagnosis and a pretty much poor outcome for patients. The only way to diagnose cancer at the moment is a bronchoscopy, followed by tissue biopsy. That's not going to change. I've spoken to doctors. That's going to be part of the process for as long as they know for now. But what I'm sort of postulating is a current, like the screening process for lung cancer. So we know that screening processes work. We've got the National Bowel Cancer Screening Program. It has 90% of cases which can be treated and detected early. Got breast cancer screening, 74,000 deaths per 100,000 in 99, down to 40 deaths per 100,000 in 2014. And in fact, Kathy, a student, is looking at a spectroscopy project to look at an earlier cancer detection for nicer cancer diagnosis process rather than mammography. But again, I don't have time to talk about that. So we've got national cervical cancer as well. 85% of precancers can be treated within 6 months. So that's where I'm coming in from, looking at a breath angle. So each of you have excelled about 20 litres of breath since I've started talking, much more since everyone else started talking. And in fact, it's about 7,000 litres a day. And it was Garima who was looking at these things called extracellular vesicles in Wellington, and they're nanoparticles, so that's why I was interested in them. I'm a physicist. I like to squish them, I like to count them, I like to see how many are there. And they're great because they're acting as biomarkers these days. So biologists look at them because they have cancer proteins. I like to look at the proteins on the surface of those, and I like to target them. So she was reading a manuscript and it had proteins there, and she said, oh look, there's some proteins there that tell us that there might be some of these nature's own nanoparticles in breath. And I said, okay. So I went into a freezer, quite literally, breathed into a tube, collected a sample, and we did some analysis and we found these nanoparticles. So that's the story. So that's what I'm thinking. thinking, well, we have roadside alcohol testing on the roads. I want to make that accessible to something like lung cancer. Very blue sky, but that's how we do research, right? We aim for big. So these are what they look like. There's Grima. So we have a living cell. Almost every single cell, this is what amazes me about biology, almost every living cell will release these nanoparticles, and we call them extracellular vesicles. Once upon a time they were called exosomes, now they're not. So it changes fluidly. But they range about, the ones that we're interested in, about 50 to maybe 200 nanometers. And so they have this lipid membrane, and they have these proteins in the membrane, and they have a lot of other stuff in there, RNA, DNA, other proteins, but we're interested in what's on the surface that we can access. I said almost all living cells release them, so we know that cancer cells do, we know that healthy cells do, so they almost are a mini-me from the cell they came from, and so that's what we're looking at. We're looking at seeing if we can get these from saliva or from breath, and to see if we can tell what cell they came from and whether that cell was diseased or not. We also look at some from E. coli cells, and for a moment there, I did look at plants as well. But let's get back to the story. I go on tangents, my students love it. So we have these pictures here. I have little spheres. That's what they look like when we look at them. So I have many nice techniques that I like to use, one being electron microscopy, the other one being atomic force microscopy. I like atomic force microscopy because it was the same year I was born that it was developed. Silly things, right? But yeah, so with the electron microscope, we look at electrons either hitting and rebounding, that's the top one, scanning electron microscopy, or we look at transmission where they go through the sample. And so these ones show the spheres that they look like directly from my breath, little nanoparticles, and then the electron microscopy gets a higher magnification, and you can see the higher dense region. This was where all the electrons are. That's giving us an extracellular vesicle, and it's about 100 nanometers. So we're looking at capturing these on a gold surface, and that's our electrode for an electrical response. So I'm going to go through that in the next part. I do that using aptamer technology. We use DNA aptamers, we have RNA aptamers too, but I don't use those yet. At the moment, we use short strands of DNA that specifically target one of these proteins. I'm going to say this one is a CD63 protein, and that's prominent on all of these extracellular vesicles. That's where we started as a proof of concept. We just wanted to see if we could capture them directly from breath. So if we have a look at, I didn't know what sort of audience we had. I knew it was going to be broad, so I just wanted to touch on what size scale we're considering, and also that way I can show you some of my quantum dots that I make. But we're looking at the 1/1000th of a metre range. So the quantum dots, for example, they range from about 3 to 6 or 8 nanometres, and just changing their size will give us a different fluorescence. So you would have heard of the QLED TVs. They have the exact quantum dots that I make in my lab, except for my quantum dots are for biomedical applications, so I can put these targeting ligands on those, and I can target something specific, like an E. coli cell or an E. coli extracellular vesicle. And it's all about to do with the physics of those. So the size gives us a band gap, we can excite it, and the size then relates to an emission that relates to the size of that energy band gap. All right, so this is the device that Zarina, who got stranded in New Zealand, made. So all in the luxury of a surrogate lab, Volker's lab, which was very nice to have. So she used photolithography, and this is a samples circle here. So we've got 4 different spots here that we can use. And so what she did, she spent 4 years of her research optimizing the attachment of the aptamer, which targets the extracellular vesicles. So she started with the target of the CD63, which is on all of them, and then she looked at CD44, which is a lung cancer biomarker, or present for most lung cancer cells. And in fact, she was able to show that it works for both. So not only did we establish a nice method to produce an optimal layer of these targeting ligands onto a gold surface, but then she could apply it to 2 different targets on the extracellular vesicles. And so she used this term called electrochemical impedance spectroscopy to measure that. And so what we'd see is we've got these they look like DNA strands because they are, but they specifically target either CD63 or another substrate could target CD44. And once you scan that with electrochemical impedance spectroscopy, we get this semicircle. So I'm not going to go into the depth of that because that would be an hour lecture, and I've already done that at 12 noon today and then jumped on a train to get here. So, and thank you for being more captivating than those students. So that just relates to the resistance of the layer. So we expect a gold surface to have low resistance because it's a nice conductor, then we add a layer and it has a higher resistance. And that's what we get. So it forms a resistive layer and we get a response. And that response is actually related to the concentration of these nanoparticles that we captured from breath. Now, this was a great start. It was the first prototype, and these are just some of the results that we're producing in this journal article at the moment. She submitted two weeks ago. So we have the optimization, so how long it took to form a nice layer. And in fact, it forms a nice layer at 6 hours, and then it goes to crap. No, it doesn't go to crap. It allows more electron flow through. We wanted a nice semicircle, the largest we could, so that way we get a better response, and we can measure that. And we can see that we've got the CD44, and we use this aptamer called AppTex. I'm pretty sure that's just because it's an author's name. And so that targets CD44. We just started with the protein. We wanted to make sure it worked for the protein first, and then we got some lung cancer extracellular vesicles, and we started there. And we did all the background controls as well. So it was great, it worked, but now we want to make it better. And there we go. And this is where my good friends Craig Priest and Ben from the University of South Australia, yes, that's where I did my PhD, so I have known them for a while, so we're upgrading. It doesn't look like much, but you can see the gold. This is one sample here, but what they're able to do is to make these little micropillars. So the scale there, I like to walk around, it's very restrictive, right, this. But this little green bar here is about 2 microns, and in fact they're about 10 microns high. And those little stripes in the background, that's our gold electrode. So they're indigitated? Yeah, I can't say that word, but it's called an IDE. That's what I've got here. So it's a gold pillared IDE. And so not only does it allow these extracellular vesicles to bind to the surface, but it stops other larger stuff from coming in and affecting it. And it's a really cool technology that we're working with, and it's a very good way to improve our device even further. And in fact, it only needs nanoliters of solution. So it's got this wicking thing to it, so all the solution gets sucked into those pillars quite nicely. So this is just some of the work that we've done where they've looked at the bare IDE. So this is without the pillars. And you can see that the normalized current density, so that's just meaning what current goes to the electrode, decreases. So it goes down to 20%. And we postulate it's because the larger tumour spheroids that we have put in the solution are blocking anything from happening. When you compare that to the pillared IDEs, we get quite a constant 100% transfer of the electrons. So it means that it's definitely anti-fouling is what we've sort of come to it. So that's what we're doing. We're getting our breast samples. And in fact, that was part of Zarina's project, but she had an ethics approval, but then, what do you know, we couldn't do it. So now Emma's taken over that ethics, and she's currently in the process of collecting 50 patient breaths, just of healthy patients. We want to get a good sample and establish a method first. And so we're going to use these little gold electrodes with the micropillars. We're going to attach the aptamers, which she's successfully done, and she's even done the preliminary work where she's attached the EVs on there as well, or the extracellular vesicles, and we've been getting some nice responses there, which is really good because they're a little bit fragile, so we wanted to make sure they worked well, the micropillars. So I wanted to cover some other applications. I mentioned the viral detection, so that's with Professor Nathan Bartlett. So he's done a lot of work in virology, and so he provided these slides for me and then interpreted them for me so I could interpret them to everyone else. But he's got this culture of cells, epithelial cells, and they relate to the air tract cells, so they're very similar. And so what we're postulating is that if we infect those cells with a virus such as COVID, which he's got a lot of experience in, and then try and look at the EVs that those cells release, we'll have an indirect method to monitor viral detection. One that we can do from breath, just like I did with the like we're doing with the lung cancer. And so we don't even know if these cells do release extracellular vesicles until about a month ago when we got this image. So we're able to wash the cell's surface with PBS, just a buffer, collect it, isolate it, and then put it onto our electron microscope to see these nice nanoparticles. And you can even, this one, you can see the nice lipid bilayer here, about 7 nanometers as expected. There's a lot of fluorescent microscopy here. I think it just relates to how the cells look here. So we've got the tubulin here, and I'm glad Nathan's not here, but he would be able to tell you what they are. And they got mucosa. Remember the mucosa in red? But it definitely reflects the cells that are just in your breath tract there, your airway. The other project is in collaboration. I have lots of collaborations because I'm a physicist and they are experts. Carl Hassan, he looks at microbiology. And so we've got 2 different projects on this where we look at targeting E. coli cells and then targeting extracellular vesicles that are derived from these cells. And we can use that as another mode of detection because one cell is a lot harder to capture than multiple nanoparticles that are quite easy to capture. And so that's what my student's been doing, and she's been using quantum dots. So we've got indium phosphide, zinc sulfide quantum dots with the targeting aptamer specifically to E. coli, and we've been using that to look at the extracellular vesicles that way. Something else I thought was fun, because I like to manipulate these as much as I can. Like I said, I like to squish them. I also like to make them explode, and I also like to make pores in the membrane. So that's what we did here. We actually put an electric field on these extracellular vesicles derived from Pseudomonas aeruginosa, another bacteria. And what we were able to do, if we had nanoparticles such as gold nanoparticles that were less than 10 nanometers, and then put those in the solution with the extracellular vesicles and then zap them, the pores would be big enough for those nanoparticles to go inside. And that's what we see here. So we've got a 50 nanometer extracellular vesicles and about 5 to 7 nanometer gold nanoparticles. And in fact, gold nanoparticles, if you ever want to do outreach, is one of the most easiest nanoparticles to make. I do those for outreach on a regular basis, so let me know if you want me to give you a nice recipe for students. But yeah, so you just use a citrate that stabilises them. And in this one, we actually used an thioaptamer for Pseudomonas aeruginosa, which was another application back in Wellington. We also did the same with our quantum dots. So we've got our indium phosphide zinc sulfide quantum dots. These ones were red fluorescent, so about 5 nanometers. And we put our aptamers on the surface in a similar way using something we like to call click chemistry. And we loaded them inside our extracellular vesicles using a similar way. And we had to optimize it. Obviously, if you increase the electric field enough, you're going to make them explode. If you don't, then they just just do nothing. So I'm on time, which is good. I have to acknowledge the students. They're the ones who've done all the work here. And yeah, the group is growing. Zarina's just submitted a PhD thesis. And in fact, I forgot to add Harleen to the mix as well. So apologies to her. She started earlier this year. And the collaborators, so we've got Carl Hassan, who's a microbiologist, Nathan Bartlett, who's a virologist, Craig Priest, who runs the AMP node at UniSA and does all the machining, all the engineering. They have 3D printers, a whole entire room of them. It's amazing. And he's postdoc Bin Guan. And Volker Nock from the University of Canterbury, who allowed Zarina to complete her PhD. Thank you again. And yeah, I'm looking forward to the Q&A.
Before we commence our proceedings for the second part of our talks, I'd like to introduce our event sponsor, Helen Lorigan, CEO for the Laboratories Credit Union, to come forward to introduce LCU to us. Please make her feel welcome.
Thank you, Fred. Good evening, everyone. It's wonderful to be here again. Last time I presented here, it was for our Community Outreach Award, and I'll talk about that in a little minute because our winner was here tonight, which is fantastic. So I'll just get my... of course, this never works when you need to work. Technology is great when it works, as I always say, which is why I bring my paper as well. It's a great pleasure to be here today on behalf of Laboratories Credit Union. We're normally known as LCU, and we're going to work with our brand, do some rebranding later in the year and become a bank, which we are. But we'll probably keep that LCU because all of our members know us as LCU and our new members know us as LCU. It's a great brand. It's wonderful to be the sponsor for this wonderful event for the Frontiers of Science. I'd like to acknowledge Dr. Fred Osman for his tireless work in arranging this forum, other Reiki events, AIP forums that LCU's attended and sponsored, and the work that he does with universities, government and the education sector to promote the higher education in science, be it physics or chemistry or mathematics. There have been a number of outstanding speakers here at tonight's events that I've been very impressed with. There were welcome addresses by Dr. Susan Pond, AM, President of the Royal Society of New South Wales, Mrs. Mary Stewart, who I've met before at the event last year, the New South Wales Branch President of the Royal Australian Chemical Institute, and Professor Miroslav Filipovic, I hope I pronounced that correctly, the Chair of AIP New South Wales. Did I? Oh, good. Mr. Mark Curray, I was very interested to hear his presentation. In my previous role, prior to being with LCU, I spent 6 years as a partner in a global venture capital firm, and we worked with a lot of the startup hubs in New South Wales and in South Australia. Both the former government here and also the former government in South Australia set up those hubs, and we're invested in a lot of startups, so that was just fascinating. So that certainly related to me. Professor John Mattick did a fabulous presentation. I very much enjoyed that. And Dr. Renee Gorham, what can I say? Renee was the winner of our Community Outreach Award last year, so congratulations again for that. After the break, we have another wonderful speaker coming up, Professor Kate Jolliffe. So he's the Associate Dean of Research and Professor of Chemistry from the University of Sydney, and Associate Professor Richard Garner from the School of Mathematical and Physical Sciences from Macquarie University, followed by the panel discussion. So now I'd like to give you some quick background about LCU. We were formed, we're sort of like a startup, and we're just really starting to scale now. We were formed in 1954, so we'll be 70 years old this year, which is fantastic. I say that because with startups, it's all about the J-curve. And so we grew very steadily, and we now really want to take advantage of all the core values that we've got. It's just a wonderful organisation and scale, and be able to provide benefits to a greater proportion of our members and more products and services. We started providing financial services to CSIRO, so we're extremely proud of that ongoing relationship, and we sponsor some of their scholarships as well. And we've maintained that relationship, as I said, for coming up now to 70 years. A large number of our members are CSIRO alumni, and ideally many more will become members. But I think equally importantly, I joined 2 years ago in May this year. We've also branched out to other groups of professionals, STEM company workers, via specialist brokers, online marketplaces and industry associations. And we also receive multiple daily inquiries about our loans and term deposits from friends and families of existing members, which is quite remarkable. Interestingly, I attended the industry forum for us is COBA, so that's the Customer Owned Banking Association. And they had an annual chair and CEO forum this week in Sydney. And so my chair, Gordana Papic, attended with me. And there are 56 member-owned banks, if you like. And a very similar thing happened with super. I used to manage the largest super fund in the country at CBA, but it was a retail fund. Now, my money's with the largest member-based super fund, Australian Super. So it's been a very interesting turnaround about values and how you look after members rather than shareholder returns, quite a different paradigm. So out of the 5 million Australians that now bank with member-owned banks, which we are one, we have $160 billion of assets. And it's growing quite substantially. The other interesting stat that I found was that in terms of customer satisfaction, out of member-based banks like us, and our stats are very similar, customer satisfaction rates in excess of 92%. Out of the big four banks, and I work for two of those, it's about mid-70s. And that's not very different to when I was at ANZ back in mid-2000s, so that hasn't changed. Our value proposition has been formulated it's all about providing the best member experience possible and service. So at LCU, that's I suppose probably the key tenet of our value proposition. So we look after members. You also enjoy low fees. We have very competitive interest rates and term deposit rates, both for our first home buyers product for our lending, and a special fixed loan product for new home purchases or refinances over $350,000. All our loans over $300,000, we have a variable interest rate of 5.95%. And we were recently, just a month ago, awarded CanStar, Australia's largest financial comparison website's 4-star award rating for outstanding value home lender for 2024. So that's just fantastic. So, and testimony to my team as well. So, and our members. We offer great term deposits on our term deposit products as well, and we can certainly work with members with discretions for larger amounts, and provide loyalty bonuses for retention, and some great kids products. So we're at the moment looking at our whole product range and really refining some of the products that just really haven't added value, and really remodelling some of those core products too, which is fantastic. The other really interesting thing is I have pretty much a whole new team. We've hired a new team over the last 18 months. 2 of our existing team members are there who are fabulous, but it's just been wonderful to regenerate the organisation with fresh thinking, experience. And I was really nervous. I thought, oh my God, a whole new team, but it's just been fantastic. So again, the results we're getting are testament to our team. And our board's done a great job too. We have a member here tonight, so I have to say that, sorry. I think the most important differentiator though, having worked for ANZ and CBA, and having recently had my ID stolen through MyGov, when I had to go to 20 institutions, when I went to my institution, they fixed it straight away. With one of the other banks, I had to wait. I still had a holding account with one. It was 4 days before they could come back to me about fraud and keeping me on the phone for hours at a time. So I kept working while I had the phone on hold. So it's a very different proposition. So I suppose when you deal with a member-based bank like LCU, you speak to a person, and often it's the same person when you call multiple times, not an amorphous call centre overseas somewhere, or to an automated phone system, or through a chatbot. And look, I've invested in chatbots in my former role. They play a part, most definitely, for self-directed transactions. But when you want to talk about something with a bit more complexity, it's really nice to be able to speak to not just a human being, but someone who's skilled and knows what they're talking about too. So tonight we placed a LCU contact request form on everyone's table, and we'd love you to make contact with us if you'd like to discuss your needs in terms of lending or any of the savings or investment products we've got with some of our highly skilled staff members. And now I'd like to actually introduce you to some of those members who are here tonight. I have 3 of my team members here. The first one who I'd like to come up and say hello is Nicole Hayway. She's our head of lending, and I'll hand over to Nicole.
Thank you, Helen, and thank you everybody for having us tonight, and we're so proud to be able to sponsor this event tonight. My background has been always in customer service, always being there to be able to help people, and I see you guys do the same thing with your research and what you aspire to achieve with what you're doing. Not the same level, but I always want to be able to help people, and that is what LCU does. We are there for our members. We are there to be able to give you products and services that are able to get you to somewhere where you want to achieve to be in a better situation in your life. So I can't do that without having a great team as well, and my team that I have, I've brought along today is Sapna. And she's our lending officer, and we also have Hannah as well, and she's our MSO/lending officer who's come in to help us today. We, as Helen mentioned earlier, we're the people that you will talk to. We're the ones that when you know that you ring up to talk to somebody, this is who you're going to talk to every single time.
All girls.
Yeah, I didn't even think, Sorry, there's 2 boys in the office and we're there to help. We're there to offer you guidance from the beginning to the end, especially in the lending. It's a big deal if you are going into buying your first home. And I know a lot of people have probably already done that already, or are looking to refinance, or are looking to go to their next stage in their life. We can take you through and on that journey as well. So can't do it without these guys, and I just want to introduce you to them, and thank you again for having us here today.
Thank you, Helen, and thank you, LCU. And this event couldn't have actually run without LCU subsidising the tickets for this event. We really appreciate your support, your kind, valuable friendship, and we look forward to continue that relationship this year and beyond. Our next presenter is Professor Kate Jolliffe. Is a supermolecular chemist who received her BSc in 1993 and PhD in 1997 from the University of New South Wales. She held position at Twente University, the Netherlands, the University of Nottingham, UK, and the Australian National University before moving to the University in Sydney in 2002 as the Australian Research Council QE2 Fellow. She currently holds the position of Payne Scott Professor at the University of Sydney, is the Associate Dean Research for the Faculty of Science and the New South Wales Node Leader for the Australian Research Council Centre for Excellence for Innovations in Peptide and Protein Sciences. She's a Fellow of the Australian Academy of Science and has been awarded the Beckwith 2004, Biota 2006, the Birch 2017 and the H.G. Smith 2018 and the Margaret Shield in 2021 medals of the Royal Australian Chemical Institute and the Royal Society of Chemistry Macrocyclic and the Supermolecular Chemistry Award in 2023. It gives us great pleasure and privilege to welcome Professor Kate Jolliffe. Thank you.
So what I thought I'd start by doing, as Fred said, I'm a supramolecular chemist, and what I want to talk about tonight is some of the work that we're doing at the University of Sydney in the general area of supramolecular chemistry. But most people don't know what supramolecular chemistry is. It's a relatively new subdiscipline of chemistry, really focused on taking inspiration from the really amazing molecules of nature. So back to amazing molecules. Nature's molecules are amazing. And they recognise each other, they form well-defined complexes, and that is really what underpins the majority of biological processes. From enzymes that catalyse reactions with amazing specificity and way better than most of the catalysts that we as chemists design, through to how viruses interact with human cells to infect us with COVID, as in the centre there, all the way through to these amazing molecular machines. The movie on the right is a protein that's carrying a cargo along a microtubule. And that is happening because the protein can step by recognising the microtubule as it moves along. Truly amazing processes that biology does. We as supramolecular chemists really want to try and mimic these processes, but with smaller molecules. The reasons being that we could then start to understand better how some of these biological processes work, but also that we can then expand out how biology functions to a wider range of applications that are outside biology. So that's really what supramolecular chemistry is. 1987 Nobel Prize was awarded effectively for the syntheses of molecules that mimic important biological processes. And that was really the birth of supramolecular chemistry. So you can see on the slide here, a molecular recognition of a potassium ion, some self-assembling nanotubes down in the bottom corner. And if I play this little movie, this is actually a molecular machine assembled by chemists from small molecular building blocks that works to form a new molecule. It makes a reactive arm that then puts together building blocks to make a completely new structure, in the same way that biological machines can make new structures, or that kinesin can move along that microtubule. So really, truly amazing small molecule machines, and this is supramolecular chemistry. My interest in this area is in the field of anion recognition. Now, anions are negatively charged ions or molecules, and they're everywhere. Our interest in this area is really inspired by the fact that molecules that can selectively detect, sense, extract, and transport anions have applications that range from the environment through to medicine. In the environment, anions like nitrate and phosphate are responsible for the eutrophication of waterways. And if we could sense their concentrations, we could work out how much of them is there, or if we could extract them from the water, that would give us a way of treating our water and removing pollution. In industry, anions are often present in feed streams and waste streams and need to be removed from those. And one great example of that is the sulfate ion, so SO4 2-. It's present in waste streams and in nuclear waste. And in nuclear waste, the presence of sulfate ions actually causes a particular problem in turning that waste into a solid, storable form. It causes the glass to be leaky. If we could remove the sulfate from the waste before we turned it into a solid, we'd be able to store our nuclear waste much more safely, and that's going to be really important as we move towards nuclear submarines in Australia. And in biology, anions are everywhere. They're the starting materials, the products, or the intermediates of pretty much every biological reaction. DNA is one big anion, for example. And so the ability to sense concentrations of anions in cells or to move them, for example, across a cell membrane, has applications in both the diagnosis and treatment of a range of diseases. But all of these applications require us to be able to selectively bind to anions in water or much more complex environments like nuclear waste. And that is actually a fundamental challenge for us as chemists, because water really likes to bind to anions. And so if we're trying to selectively sense an anion in water, we have to displace all the water molecules that are around that ion in order to get our selective binding to occur. So inspired by nature, we look at how nature binds to anions and try and mimic that in our small molecules. I've got two examples here to show you how we do this. So the first is the sulfate ion. I've got here the structure of the sulfate binding protein, so the ribbons and the curls of the alpha helices and the beta sheets in the protein structure. Right in the middle of that, you probably can't see it, is a sulfate ion. If we look deeper into that structure, we can see that that sulfate ion is bound in the middle of that protein through a network of hydrogen bonding interactions. And I've drawn those out in cartoon form and also in the chemical structure form so that you can see where those hydrogen bonds come from. They come from the backbone of the amides, the peptides, and they come from side chain hydrogen bond donors of the peptides. So if we could array some hydrogen bond donors in the right geometry, we might be able to selectively bind to sulfate ions. Similarly, this is the alkaline phosphatase from E. coli binding to phosphate ions. Now, in this case, you can see again a network of hydrogen bonding interactions, but also here, there are 3 metal ions that I've circled in green so you can spot them. They're a bit hard to see. And these metal ions also bind to the anions because, of course, metal ions are positively charged, cations. Anions are negatively charged, opposites attract. Those metal ions help to bind that phosphate ion and also to catalyse reactions in this particular case. So again, here's another interaction that we can start to exploit in our small molecules to interact with anions, and I'll show you how we do that in a minute. One thing that I should point out is that anions come in a variety of shapes and sizes. They can be spherical, things like chloride ions. They can be linear. They can be tetrahedral, like sulfate and phosphate. They can be octahedral. But the key thing here is that they're often really quite large structures in comparison to cations and just to atoms. So you can see here the ionic radius of the fluoride, chloride, bromide and iodide ions is about twice that of the elemental atoms themselves. Now, we're talking, 0.2 nanometers here. much smaller than any of the structures that we've seen already this evening, the quantum dots. We're going much, much smaller, but to me as a chemist, these are actually quite big structures. And in order to bind them selectively to array our interactive hydrogen bonds or metal ions around these anions, we need to create large molecular architectures that match the size and the shape of the ions that we're interested in. So something that matches the spherical nature of a chloride or the tetrahedral nature of a sulfate or phosphate ion. To do that, we use the tools of organic synthesis. And in this way, what we do is we take building blocks, a bit like these Lego pieces here. They might be atoms, they might be molecules, they come in different shapes and sizes. And in the same way that I can put these Lego blocks together in different ways to make a range of different architectures, I can do the same thing with my molecular building blocks and take hydrogen, carbon, nitrogen, oxygen, along with other elements, put those together by forming new chemical bonds between them to make an array of different molecular architectures that are targeted to the size and shapes of the anions that we're interested in binding. And so you can see here a selection of the sorts of molecules that we make. For the chemists among you, those structures will be interesting. Everybody else is probably going, The lines are bonds between carbon atoms, and we don't draw the hydrogen atoms in generally, so you don't get to see those. These different molecules have different potential applications, from extraction of sulfate ions from water through to sensing pyrophosphate ions and phospholipids on cell membranes. Of course, when I think about playing with molecular architecture and building bonds between atoms, you're probably thinking of me sitting in a lab with a tiny weeny pair of tweezers and taking atoms and putting them together. That's not how it works. Everything we do is done on the bulk scale. Of course, if you think about a litre of water, or 22.4 litres of water is rounding 6 by 10 to the 23 water molecules. So, that's a lot of atoms to play with in a small flask, right? So everything we do is done on a bulk scale when I think about chemistry, but actually we're making milligrams to grams of compounds. And of course, those can then be used in applications. So we mix things together a bit like following a recipe sometimes in the kitchen to bake a cake, but here we're mixing chemicals together just like the ministers or shadow ministers' chemistry set. We don't tend to use Bunsen burners anymore, in order to make those reactions between molecular species occur to build the structures that we want. So I've got just a couple of examples for you in more detail of what we've looked at. The first example I'm going to look at is some sulfate extraction and transport work that we did. And we created these quite interesting looking molecules called squaramides. And they're called squaramides because you can see that in their structure there's a square. It's a cyclobutene ring. It's really highly strained but surprisingly stable under these conditions, and really good. Those NHs attached to it are really good hydrogen bond donors, super powerful hydrogen bond donors. If we can put those in the right positions, we can start to bind to anions, and we can start to do it selectively. And in this case, I've shown two structures that are similar to each other, but one's a little bit bigger than the other. The small molecule binds to sulfate ions, but it also binds to a range of other anions, phosphate ions and acetate ions, for example. And that's because there isn't a very good match between the size of the ion and the size of what I'm going to call the molecular host. The larger structure there, what I call the 3+3 macrocycle, it's got 3 squaramides in it, perfectly fits around a sulfate ion. So you can see the space filling model on the right-hand side of the slide. That sulfate is kind of bound really nicely. There's not much gap in the middle of the structure. The hydrogen bonding interactions are set up perfectly to bind to those oxygens in the sulfate. And the sulfate is a little bit protected from the water that might be around it in solution by that molecular structure. And that allows us to actually bind to sulfate ions in aqueous mixtures and to extract those sulfate ions from an aqueous phase into an organic phase, so a chloroform phase in this case. Now, this is an example of where a proof of principle experiment where we could imagine using this type of molecule to extract sulfate from nuclear waste. So we have set up a little U-tube here. We've got water in the two arms, and in between them we've got chloroform. And chloroform and water don't mix, right? Like oil and water. Chloroform sits underneath the water layer. It's heavier than water. We put in the source phase here some sodium ions and some sulfate ions. And if we do that and we look at what comes out into the receiving phase, over 21 days, we see nothing. That's the purple line at the bottom. If we put our macrocycle, our 3+3 system, into the chloroform layer, again, after 21 days, no sulfates come across that membrane. But if we add a little bit of another anion, nitrate ions, that bind less well into our macrocycle, then we start to see an exchange between those anions and the sulfate is then released out into the receiving phase. And that's the green circles there. So after 21 days, we're starting to see sulfate appearing in that receiving phase. And we can actually speed that up by adding something else into the receiving phase that helps to remove the sulfate as it's put in, and that's barium ion. So barium sulfate precipitates out of water. It's got very low solubility. And so you can use the barium to precipitate the sulfate out, removing it out of that water so that your equilibrium allows you to bring more sulfate through. So this is just a really simple example of how we could use these sorts of molecules to extract sulfate from an aqueous solution. You can imagine in that source phase that being nuclear waste, completely separated from the outside world by that membrane of organic material. That's an extraction application. The other applications I want to talk about are in the sensing area. So again here what we're trying to do is to selectively sense one particular anion over a range of others that might be quite similar in size and shape and charge. And the idea here is really very similar to that where we look at enzymes, where they, that lock and key model that's often talked about for enzymes, where the substrate fits beautifully into that enzyme site. What we're doing here is building a molecular recognition site that fits best to the thing that we're interested in binding. And so only what we want binds into that site, leaving all of those other shapes that don't fit in that hole outside. Attached to that, though, we need to add something that tells us when that binding event has occurred. So that might be something that changes colour, or it might be something that lights up by fluorescence when a binding event occurs. And this is kind of like the dipsticks that you have in your pool where you check the chemical levels. You're just putting one of those in, it changes colour and you know what the chlorine content is or something like that. Exactly the same principle here, but now looking at anions. So here we have an example where we're looking at binding to pyrophosphate up there in the top corner. And this is a molecule that's generated in many biological reactions in the presence of many other similar phosphate derivatives. The ability to sense this allows us to monitor enzymatic reactions, but it can also be useful in monitoring pyrophosphate concentrations in physiological fluids like urine and synovial fluid, where the presence of different levels of pyrophosphate is a marker of a number of different diseases. Bone diseases, arthritis, those sorts of things. So you can see here, again, this is now a fluorescent system rather than a colour change. If we take our molecule, and again, I've put the molecular structure up there for the chemists in the audience who care. We've got the fluorescent spectrum. As we add pyrophosphate into our molecular sensor solution, we see a turn on in the fluorescence signal. And that's projected out on the graph on the right side, where you can see the pink dots belong to pyrophosphate. As we add pyrophosphate in, we see a turn on the fluorescence. But if we add other biological phosphates, things like ATP and ADP, we don't see any change in fluorescence of the system. And so this is a nice selective sensor for pyrophosphate. It turns on with pyrophosphate, but not with other biological phosphates, allowing us to monitor pyrophosphate in mixtures. And the final example that I'd like to share with you is something that we're really focusing on at the moment, and that is sensing differences in cell membrane compositions. Now, we've seen a couple of examples of membrane bilayers in the previous talks this evening, and you can take one single phospholipid molecule, if you choose the right one, and it will form a bilayer membrane. There's no reason for biology to use the thousands of molecules that it uses to form cell membranes. There are over 1,000 different lipids in your mammalian cell membranes. We don't really understand why there are so many of them, or indeed what they all do. And that's because biologists don't have tools that allow them to look at what those phospholipids are in real time. So membranes are really dynamic environments. They're changing all the time. And the composition of the membrane changes with different stages of the cell life cycle. It's different in different cell types. It's different in different organelles. And it's not symmetric between the outside and the inside layer of the membrane. So being able to look at changes in that membrane environment is something that would be really important in understanding biological processes. So I've shown a couple of examples of the head groups here, and you might be able to pick out that some of these are anionic head groups, some of them are overall zwitterionic head groups, but we're really focusing in on those anionic systems. One of which is shown here in red. So this is the head group of phosphatidylserine. Now, in a normal mammalian cell, most of the phospholipids are zwitterionic, and I've shown the structure of the most common ones on the slide. Phosphatidylserine is anionic, and in a healthy mammalian cell, it all sits on the inner leaflet of the membrane, so projecting towards the inside of the cell, there's none on the outside, effectively. But when a cell undergoes cell death processes, and that could be apoptosis, programmed cell death, that phosphatidylserine, that anionic lipid, flips to the outside layer of the membrane. So an anionic cell surface, in a mammalian cell at least, is a signal of cell death, and it causes lots of biological processes to occur. So, macrophages know to come and clear that cell away by that anionic charge. So if we can detect that anionic charge on a cell surface, we can start to monitor cell death processes. And that's what's shown in the cartoon on the side there, is the molecule that we're going to make that's going to come along and only turn fluorescence on when it binds to an anionic cell surface. And we can demonstrate this we demonstrated in lots of models, but then we moved into cells, and this is a cancer cell line. And we can see in this example that if we take the cells and we just look at them under the fluorescence microscope, in the channel in the middle, the green channel is our probe, we don't see a lot happening. There's not much going on there. But if we make those cells die by adding a cancer drug, in this case camptothecin, you see that beautiful green halo around the outside of the cell where the cell membrane would be. And this is showing us that we're recognising that anionic charge, and it allows us to now follow that cell death process in real time. So here are some different cancer cells, again treated with the same drug. Over time, you can see they change shape. That's because they're dying. But if we look at the fluorescence microscope, we can also see, even visually here, that there's an increase in fluorescence, and we can plot that by mean fluorescence intensity, showing that the cells stay alive for 3 hours, and then over the next 3-hour period, they all die. If we do the same thing, but we don't add the cancer drug, we don't see any change in the fluorescence of those cells. So we're now able to monitor that cell death process using what's effectively an anion receptor. So those are some examples of where we're trying to apply anion recognition to problems in biology and the environment or industry. There are lots of others, but I don't have time to talk about them tonight. So I want to finish off, though, by saying that also, it's not me who does the work here in the lab, and not anymore anyway. It's a group of fantastic PhD students, early career researchers and honours students who work with me and have worked with me over the 20-something years I've been at the University of Sydney. And actually, for me, their development as scientists and researchers is way more important than the outcomes of the research that we do. Training the next generation of scientists is something that is super important in my job, and in fact the best part of my job, I would say. So I need to thank all of them. Their names are all here, the current research group on the slide, and I need to thank the ARC for lots of funding over the last few years, and the University of Sydney, and thank you all very much for staying after the little break.
Thank you, Kate. That was excellent. Haven't our presenters so far been outstanding? All complement each other's research, and we're very lucky to actually go to our final speaker, which is our maths and computer science background. Associate Professor Richard Gardner is an Associate Professor of Mathematics at Macquarie University, Sydney. He grew up in the UK, received his PhD in 2006 from the University of Cambridge, and spent some time on the postdoc circuit in Europe before joining Macquarie in 2011. He still says whilst rather than while, but has at least learned to use the word heaps. His research is in the mathematical area of category theory, which looks for patterns appearing across different areas of mathematics, theoretical physics and computer science, and creates new languages for studying them. This research has been recognised by, among other things, his award of the Medal of Australian Mathematical Society in 2017. He's also a keen teacher and has tremendous fun explaining discrete mathematics and calculus to first and second year computing and engineering students. Last year, this was recognised for his receipt of the Vice-Chancellor's Student Nominated Teaching Award. Ladies and gentlemen, please welcome Associate Professor Richard Gardner.
Thank you very much. And I've realised I need to go and get a little cable out of my bag, so I'll be back in about 30 seconds. You want this one? Is that good? Nah, that's not the right one. There we go. Okay, I like to keep you waiting. I'm going to take the other end and plug that into there. And hey presto, we're on the wrong page. All right, let's start at the beginning. Okay. Does this microphone work? Yes, does this microphone work? Oh, there we go. Okay, so at least I can not be completely stranded behind the lectern. Okay, hello everyone. Thank you for all being here, and thank you to the organiser of this event, especially Dr. Fred, who's done a very fantastic job in putting all of this together, and our other speakers who've been fantastic, and it's been a very enjoyable evening so far. So I hope you're all feeling relaxed and refreshed, and not yet tired and emotional. So I'm here as a mathematician, so I guess maybe things are slightly harder for me, because I don't study very small things. I don't even study very large things. I just study things that don't exist at all. And so somehow I have to work out how to tell you some stuff about mathematics in a way that I can kind of live with myself about. And I decided the only way I could do this is if I actually taught you a bit of maths. So in my notes, I've got a little note that says, pause for Stampede to bar. But that seems that hasn't happened, so I'm very pleased about that. And so, okay, so what I'm actually going to do is I'm going to sort of come at the maths quite slowly. I'm going to start by asking a really important question, which is not that related to maths. So why do computers consistently do the opposite of what you want them to do? So I'd like to ask a question. If you'd like to put your hands up if you've sworn at your computer in the last week. So I actually haven't sworn at my computer. And the reason is that my wife taught me this amazing trick, which is that if your computer annoys you, you should hiss at it. It's amazingly satisfying. You just stand there and you go, And so I've been doing that quite a lot the last week. And so all of us have this issue, and we might ask, why is this? What's going wrong? Whose fault is this? So there are three possibilities, right? It could be that it's your fault. It could be that it's the computer's fault, or it could be that it's someone else's fault. You can tell I'm a mathematician because I've enumerated all possible cases now. Okay, so if we were, say, 100 years ago, then you could say with almost certainty that what went wrong was to do with the computer. Okay? So there you can see some vacuum tubes. So they're either very fancy light bulbs or very primitive transistors, depending on your perspective. And so they were what the first generation of computers ran on, and a typical computer would have like 6,000 of these valves in it, and they'd kind of blow once every few hours, if you're lucky, maybe once every half hour or something like that. And so you're just starting doing a calculation, then it would kind of all blow up, and you'd have to go and search through all the valves and find the one that had blown and replace it. Okay, so computers are a lot more reliable today, and so I guess we're pretty fairly certain that probably there isn't a transistor that's blown up inside our computer if it stops working. I mean, it does happen. You can zap it with an electrostatic shock and blow it up. That is possible. Or it can get struck by lightning. Or perhaps it could be a different kind of fault, like the San Andreas fault. So it's quite remarkable if you think about it. Wouldn't it be a really bad idea if you took the foremost concentration of computing knowledge and expertise and placed it on top of a highly geologically unstable area? And so I think a lot of people are just waiting for the crunch event, after which we'll suddenly be no longer be able to buy anything online ever again, because it'll all gone down a big hole in the middle of San Francisco. Okay, and so the other option is that it's not the San Andreas fault, it's actually your fault. And maybe I'm being a bit harsh on there, right? Okay, it might not be actually your fault, it might be someone's fault. Okay, and so this is normally what is the case. It's not the computer's fault, it's someone's fault. Now, quite often, it's not actually your fault, it's the fault of whoever put together what's running on the computer. Okay, and so there are various aspects in which what's running on the computer can go wrong. So let's maybe try and work out what those aspects are. So to motivate this, I'm going to show you some advice that I found when I was furiously searching on the internet how to give a public talk. And it said I should explain what I'm going to say, and then say it, and then explain what I just said. Okay, I think this is bad advice, because it's basically telling me to treat my audience like they don't really know what they're doing, okay? And I think you all know what you're doing. See, I also learned that you should compliment your audience when you're giving a talk. So this is bad advice, I think, for a public talk, but I think it's quite good advice for writing a computer program. So this is how you should write a computer program. First of all, you should describe what it's going to do. Then you should write it, and then you should check that it does what it said. And so all these three aspects have names, right? So this would be called specification. This would be called implementation. And then this would be called verification. Okay, sounds simple, right? But the thing is, in all of those stages, there are various ways in which this can go wrong. Okay? So let's have a look. So this is how not to write a computer program. So step one is to describe what it's going to do, but do it really badly. Okay? So you're going to tell people you're going to either do it in a sloppy way that doesn't specify completely its behavior, or you're going to do it in a way that's simply not what you actually want. Okay? And so you would call this, I suppose, a specification error. So the next thing you could do is, you're a bad computer programmer, you take your specification and you just type a load of rubbish that the computer doesn't even understand. Okay? So your computer has a language, and that language you have to type following certain rules, and maybe you don't follow those rules. And so if you do that wrong, you'll invoke a syntax error. The computer will complain loudly, and your program will collapse in a heap of smoke. Okay, now you may get past that point. Okay, so your specification is correct. Your programmer has correctly typed in code, and the computer accepts the code. However, it may be that the code does not do what it was supposed to do. okay? So maybe your code, maybe your specifier told you that they wanted you to add two numbers together, and so you wrote a program which took those two numbers and they multiplied them together, and so you give that back to the specifier and say, I wrote your program for you, and he looks at it and goes, great, and if he doesn't check what you've given to him, then he's going to go away and be terribly disappointed when he tries to add two numbers together and submit it for his exam, okay? And so this is a logic error. So it's a program which looks okay, but in fact does not do what it's supposed to do. Okay, and so the problem is now, how do we avoid logic errors in a computer program? And there are various levels at which you can do this. And so here we go. So level 1 is you take it and you test it under normal use conditions, okay? So you've got something that's supposed to be a word processor, and you go and you process some words in it, and it seems to do what it's supposed to do, and you, okay, great, thumbs up. So this is what you're supposed to do, which is to test it under abnormal use conditions, okay? So imagine you've got something that's like controlling an aeroplane or something. Most of the time, the autopilot just sits there and it kind of lands your plane for you and everything's okay. But then, of course, the situation is what happens if a bird flies into your engine or something like that. Then you want to make sure that the automated control system responds appropriately to that situation as well, right? So you should test not only under normal conditions, but under all conditions you can think of. Now, the thing is, you may not think of everything, okay? So there's always some even more twisted way in which things can go wrong than you can imagine. And so the ultimate, if you like, is to prove that your program is correct using mathematics, okay? And so that's what I'm going to tell you a little bit about. Is it terribly embarrassing? What time did I start? 12 minutes. Okay, great. Perfect. Okay, so I'm going to tell you about the practice. I'm going to tell you about the theory. And we're going to concentrate on the theory because that's where I'm at, I guess, about the theory. So the practice is, so this is a project which I just wanted to promote a little bit. So this is an Australian project. It's called SEL4, okay? And so what this is it's a verified microkernel. So what does that mean? So a microkernel is the thing that controls the very basic level of a computer system, right? So just the absolute basic level that interfaces directly with the hardware and kind of, right. So if you think of your operating system, it's way below that. Like it's right down, sort of just above the chip level, basically. And so to say that it's verified means that they've written a complete specification for what it's supposed to do, which is kind of a completely rigorous thing. And then they've used a computer program called a verification program to check mathematically that everything that their code is supposed to do is actually done by their code, okay? And so there's absolutely no way in which the program can deviate from the designed behavior because it's mathematically proven to follow that behavior, okay? So this project is called SEL4. It was developed by a group who used to work at CSIRO at Data61 called the Trustworthy Systems Group. So I'm not quite sure why, but in 2021, CSIRO decided that they were no longer going to host the Trustworthy Systems Group. But fortunately, they live on New South Wales, and here are some of the members. So some of my former PhD students have worked with them and had a really good time. But I'm going to concentrate on the theory, okay? So the theory is the following. Okay, so I've listed here a couple of things, which I'm not really going to talk about. So this is type theory. So this is the BDI of Per Martin Löf, who is a Swedish philosopher and logician and mathematician. And so he's responsible for something called type theory, which is basically a mathematical framework for studying programming languages and also for in the same language for kind of proving things about them. So it's sort of fascinating sort of mix-up of the computing and the logic you need to sort of verify that the computing does what it's supposed to do. And so that's that's the kind of framework in which this project was done. So getting a little bit further towards kind of pure mathematics, there's something called domain theory, okay? So this is Dana Scott, who originated domain theory. So he was a professor at Carnegie Mellon University in the States. And so domain theory a domain is like a mathematical model that represents a kind of domain of computation, you might think, and sort of the world in which computations can happen. So I'm going to be a bit vague about what exactly that's supposed to mean. But it's a sort of major way in which we can represent computation mathematically. And then the final one is the one I'm going to talk about today, which is games. Okay? So you've probably all heard of John Nash and his kind of economic games and the prisoner's dilemma and things like this. Okay? So these are quite a different kind of game. So those ones are kind of very raw and bloody and kind of, they're all about who wins and who loses and how much do they win and how much do they lose. So the games we're going to consider are a bit more like Sports Day in that it's not necessarily the winning or losing that's important, it's the taking part. And you'll see what I mean in a minute. Okay, so I'm going to start with a very boring game. In fact, the world's most boring game. So you've all played noughts and crosses, or tic-tac-toe, or whatever you want to call it. So this game is going to be tic-tac-toe, but on a 2x2 board. Now, to make it slightly less boring than it might be, I'm actually going to make it so that you can't win by making a diagonal line, because if you do that, then it would just be incredibly boring. Okay, and so what we're going to do is we're going to take turns in playing this. Okay, so maybe, does someone want to play this game with me? Anyone? Top left? Over here, someone? Yeah, okay. All right, do you want to go first? Why don't you go first? Top right. Okay, top right, there we go. Okay, so you're going to go top right. So then I'm going to go here. Where would you like to go now? Bottom left? Okay, and then I'm going to go bottom right. And there you go. So we've managed to force a draw in this game. Now, of course, if you had said bottom right instead of bottom left, then you'd have won. But you're being very polite and thought it was probably a bad idea to beat the speaker. However, in fact, the person who goes first can always win this game. And so that's a feature of the games we're going to have, that the first player can always win. And in fact, we can write out all the possible ways that the first player can win. And so here we are. Okay, so you probably can't see this, but fortunately I can do this. So this is the game tree. So we start here. And so what happens is, first of all, the opponent, as his opening move, just draws the board down. And so then this person, who I'm going to call player, who plays in purple, gets to play a circle as their first move, and they can do that in 4 possible ways. And then I, as the opponent, can respond in those 3 ways, like that, and then so on. And you can see here, in these ones, the first player will win the game, and in these ones, it'll be a draw. Okay? And so most of the time, the first player can win, but sometimes there can be a draw. Now, what we're interested in are ways in which the first player can force a win, and we're going to call that a winning strategy. And so basically, we can illustrate that just by sort of highlighting a bit inside this tree. Okay, so here is a strategy for the player who's playing O. Okay? So it says we start down here, and then I'm going to start by playing top right. And then if my opponent plays top left, I'm going to play bottom right. So that's what you decided not to do. You said, let's play up here. And then we got to here. But if you had been a bit more John Nash about it, you'd have played that, and then you'd have won. Okay, and then, but if instead I played down here, maybe we choose to do this, and so on. Okay, and the point is that we choose a thing such that no matter what move the opponent makes, we respond in a way such that we're going to win. Okay, so that's a real-world game, but we can turn that into a sort of more mathematical thing just by forgetting what game it is we're playing, and we just keep this picture. Okay? Right? It's just like, here's the first move, these are the possible responses, these are possible responses to that, and you say, okay, well, how do I know when I've won? Well, the player who plays last always wins. That's the new rule. Okay? So here, purple wins because there's no responses by the orange player. Okay? So there we go. There's our first mathematical game. Okay. So this actually leads into the first thing in my title, which is computer games. Okay? Because we can do this, not for this game of tic-tac-toe, but we can do it for more complicated games. So if you do it for 3x3 tic-tac-toe, there's 256,000 of these nodes instead of about 50. if we do it for chess, then it's astronomically large, like the number of entries is bigger than the number of atoms in the universe. And in fact, the number of entries, if I just take the first 3 levels, the moves in the first three moves in a game of chess, there's over 140 million entries there. So it's a much more complicated game, and we don't, as yet, know a winning strategy for it. And so we have to approximate. Okay, so we say, well, we don't understand the whole tree, but we kind of say, well, what's my best strategy, given that I can only see maybe 5 moves ahead, like this. And I'm going to estimate for each of the moves 5 layers ahead how good they are. Okay? And so this was actually first described by Claude Shannon in 1950, the idea of a computer playing chess. It then took about another 45 years or something like that before computers actually got really good at chess and could beat humans, and now they routinely do it, and even more recently they've become so good that they can beat computers at Go as well, which is a much more complicated game. However, that's not what I want to talk about. I want to talk about using these games to actually represent computing. So I've got 5 minutes left, and I'm going to be able to tell you just a few more slides about how we can use these games to actually do computing, to represent computer programs. So here are 2 really boring games. Okay? So in this game, the orange player starts by asking true or false, and then the blue player responds by either saying true or false, and then they've won the game. So this is a really bad game. That's why I say it's not the winning or the losing, it's the taking part that counts. Because the player who plays blue has 2 strategies for this game, right? They can either say true, I win, or they can say false, I win. But those 2 strategies give you information. It's like they're saying either true or false, okay? So the strategies for this game encode the 2 Boolean truth values, true and false. Similarly, for this game here, the strategies that the player has, the blue player has, is, so the orange player says think of a number, and then the blue player just responds by saying a number between 1 and 10, okay? They're always going to win, but each of those winning strategies encodes a different number, which is their response. Okay, so we're just going to try something a little bit more complicated than that. So I'm going to show you a construction. It's going to take some of these game trees, 2 of these game trees, and you turn them into a new game tree. So here's a game tree, and here's another game tree, which I've helpfully shown in pink and blue. And what we're going to do is we're going to stick them together. So we're going to take the blue one here, and then we're going to stick the pink one like this. But you see, I've taken the root of the pink one, and I've sort of stuck it onto the root of the blue one. And you'll see what happens is all the colours here reverse. So the roles of the player are interchanged in this game here. So that might seem like a weird thing to do, and you might wonder why on earth we're doing this. And I'm going to show you. So we're going to use the 2 games we had here, and we're going to apply this construction to them. And we're going to construct a new game, which is called Do You Like My Number? Okay, so you shouldn't confuse this game with Do You Want My Number? That's a different game. So the game Do You Like My Number? So the orange player says, do you like my number? And then the blue player has 3 choices. They can either say, yes, I do, no, I don't, or they can say, what is your number? So then, if they say, what is your number, then the orange player tells them what their number is. And you might think at that point, well, that's a very boring end to the game, because now the orange player seems to have won, and we don't have an answer to the question of whether the number was liked. However, we can actually do this because there's a new rule here, which is that we're actually allowed to jump back to earlier parts of the game. Okay? So once we've answered this question, we can then jump back to here and answer the original question, which is, do you like my number? Okay? So a typical strategy for this game might be, as the blue player, the orange player says, do you like my number? I say, what's your number? And then maybe if the orange player tells me an even number, I say, yes, I like your number. So if I get 2, 4, 6, 8, or 10, I'm going to reply with true. And if I get 1, 3, 5, 7, or 9, I'm going to reply with false. Okay? So you can see that one of the blue player's strategies for this game encodes the idea of checking if a number is even or odd. Okay? And I can think of a different strategy that checks if a number is below or above 5. Okay? And I can think of an even other strategy that just allows me to pick out one of those numbers, okay, which is maybe I decide I only like the number 3 and I hate all the other numbers. So I'm just going to say yes if you respond 3 and no otherwise. Okay. All right. So if you'll allow me just 2 more minutes, I'll show you the final slide because I think it's fun. It's all right. You can stop me. It's not that fun. So this is a game called Guess the Number. And so what do? We take this game, Do You Like My Number? And we take the game, Think of a Number, and we stick them together. And so what we get is a game where the player says, can you guess my number? And then this player says, well, hang on, what's going on here? Sorry, I've just had a sudden moment of unclarity. Okay, so this game is called Guess the Number. So this player says, what's my number? And then the other player says, well, do you like my number? And then he says, well, what is that number? And now my moment of unclarity, unfortunately, seems to have become all-pervasive, which is very sad because this is a really exciting example to end the talk with. But I think I'm simply going to have to declare that I've managed to actually confuse myself, which is a shame. But in 10 minutes, I'll be able to tell you how to play the game of Guess My Number using this game tree. And I think I'll stop there on that real damp squib. Thank you.
Hello, we're up to the question and answer period of the night. Hello, I'm Ian Wolfe. I produce Diffusion Science Radio, a weekly science and technology podcast. All these people have been interviewed for the podcast, so you can hear them again saying things in a conversational way. But if you've got questions that you've been thinking about and wondering since you've been listening to all these amazing talks, this is going to be your opportunity to ask those questions. So Fred will have somebody come around with a microphone and he'll point to people and you'll get your chance. So before I go to the audience, I've been asked to just ask a few little questions to sort of seed things. So going in the order here that we spoke, John?
Yeah.
I want to ask you, do you think there could be another foundational extrapolation big mistake in biology that's just gone? People assume something like they did with junk DNA, that they could extrapolate from bacteria, and they didn't question it, and then when people did, they didn't listen. Mm-hmm. Do you think there's another big mistake like that?
Inevitably. I was saying to someone earlier that there's an apocryphal, probably untrue story about Lord Kelvin, who in the 1890s reputed to have predicted the end of physics. He said, we've got Boyle's law, we've got Ohm's law, it's all over. We've done that, folks. And of course, then along came Rutherford and Einstein and Schrodinger and threw all the cats up in the air. Plate tectonics, continental plate tectonics, was ridiculed for half a century until the penny dropped, So the mistake that science makes over and over again is to think that it has the conceptual framework correct, rather than thinking that it may well, and usually is, incomplete in some facet that wasn't expected. So I don't know what it is, but I think science, most of science is what now the chap who wrote The Structure of Scientific Revolutions, Thomas Kuhn in 1962, he portrayed science as lurching, that's my word, from paradigm shift to paradigm shift, between which people did what he called normal science. So they designed their investigations and interpreted their results within the reigning conceptual framework until anomalies occurred. And the anomalies would be ignored until they accumulated to such a level that people said, hey, we've got something wrong here. It's usually not wrong, it's just incomplete. And you could think of Newtonian to Einstein in physics, or Ptolemaic to Copernican astronomy. So what I try and instill in my students is curiosity and to really question things that don't make sense to them. Now, it's often because they don't know enough, but I said, look, follow your nose. But you get to a point, and the more you learn as a PhD student or a postdoc, you move towards the front of the bus. You get to the front, and it still doesn't make sense, then you know you've got something. And I like a quote from Leonard Cohen, the cracks are where the light shines in.
Very good. And so, Renee, you mentioned about an indirect way to detect coronavirus with these extracellular vesicles. So the question is, do viruses not put out their own extracellular vesicles?
Yeah, well, that's actually a really good question, and we have discussed this with my good friend Nathan, but they're about the same size as extracellular vesicles, and their structure is a lot more simpler. So they could, and we wouldn't mind looking, but we reckon it would be more beneficial to go for the extracellular vesicles that come from infected cells, not only for detection, but also for the fundamental knowledge. At the end, I'm always going to be a researcher. I don't care about fundamental commercialisation, and my IP department at the university will hate me for that. They do already, because I say it all the time. But yeah, so it's more the fundamental knowledge that we get from the pathway to get to the commercialisation is what I'm interested in. Yeah.
Fantastic. We'll pass them along. So, Kate, I wanted to ask you, I was very interested in the leaking of the glass that holds nuclear waste and that we might be able to improve things. Is this something that ANSTO knows anything about?
Yeah, so we've had a few discussions with ANSTO, and they certainly think it's worth doing. Of course, Australia doesn't currently have large amounts of nuclear waste. It's a much bigger problem at the moment in the US, where they have a site called Hanford that has, I don't even know how much, huge numbers of drums that are, getting a bit leaky. And they've, had to change the pH to try and stop them from corroding on the inside. So they used to be acidic and now they're really alkaline. It's a much bigger problem in other places in the world, but the more that we start thinking about changing energy sources, about using nuclear engines to power things like submarines, then the more important it's going to become.
Fantastic. And Richard, I don't know if your 10 minutes has passed, but did you work out how to successfully play Guess My Number?
Yes. It's all totally clear in my head now.
Please tell us.
I might need to draw some pictures. So the point is that game basically allows you to develop different search strategies. So you've got a thing which takes a number and says true or false, and then maybe you're trying to find the first number for which that thing becomes true. Okay? So it's like, okay, I could ask, is it true for 1? And if it is true for 1, you say the answer's 1. And then you could say, is it true for 2? And if it's true for 2, then you can say the answer's 2. Now, there are other search strategies, right? So I could just keep going up from 1, 2, 3, 4, 5. But another search strategy would be to bisect. So I've got 10 things. So I say, is your number 5? And then if they say no, then you say, is your number 7? And so then you kind of go up or down, right? And so the point is that different strategies for this game would implement different search strategies for playing Guess Your Number or something like that. And so just starting from this very simple thing, I guess, I mean, you can see it on the page, it's not super hard, right? And just within that, there's enough richness to encode all these different kind of quite complicated and subtle computer programs for doing something as useful as kind of implementing a search strategy or something like this.
Thank you. So now we turn to the audience, and we've got one over here. We need to do it. Oh, do you want to take that one or this one?
Take your pick.
Take your pick, take one, and I'll
Thank you.
pass it along to whoever.
Hi, Professor John. You said that muscles, bones, etc., they are programmed during the conception. When you say that, I'm wondering what about before conception, when there is still in sperm and ovum? Why not? Because we do pick up that mother's trait and father's trait, that bone structure and all these things. Why not before conception, and what happens? Thank you.
That's a very good question, and I was just trying to think how to answer it. There is a lot of evidence that there is transgenerational epigenetic inheritance. In other words, we inherit both the hardwired stuff in our DNA, but softwired stuff, which turns out to be RNA mediated. And it's Lamarckian. So Lamarck, who wrote a century before Darwin, actually invented evolution. Darwin didn't. What Darwin did was give a plausible mechanism for how it works. And Darwin said that he didn't mind what Lamarck said because he didn't know what caused the variation upon which selection acts. So Lamarck, was the sort of stretching giraffe neck sort of thing, use of a particular feature, was. And the theoretical biologists in the 1930s ruled out Lamarckian inheritance when they tried to integrate Mendelian genetics with Darwinian evolution. And they said all mutation is random, which is incorrect, but they just ex cathedra said that. And they said that Lamarckian type inheritance of experience didn't exist. Again, ex cathedra experiment. And there was a bizarre experiment quoted where mice tails were chopped off for 5 generations and didn't change the length of the tails in the next generation. So they said, aha, there's no memory of experience. And when I talk about this to students, I hop across the stage like, 3 blind mice getting a tail chopped off, stupid. So that became law with no evidence whatsoever. However, and I won't go too long, 1915, an English geneticist called Bateson reported what he called bizarre patterns of inheritance, rogue patterns of inheritance, he called it, which was the first observation of what later became called paramutation, which has been really well described in plants because you can do the genetics at high resolution. There's no question that both the experience of plants for stresses or environment is transmitted to the next generation, and it's RNA mediated. The same thing happens in animals, but the extent of it is unknown because it's very hard to test because the genetics is much more difficult. But a clue comes from tomatoes, of all things, where the transgenerational epigenetic inheritance is mediated by what are called microsatellites, which are the things that you use for DNA fingerprinting in humans. And we've got about 1 million of them. And there's a lot of evidence now that we are, from studies in mice as well as humans, that we are inheriting some degree of memory. Now, the extent of which we don't know. If we take the clue from plants, it's probably RNA mediated through eggs and sperm. Some of the molecules are starting to be identified, but it's early days. And, it's one of those examples where people just made an assumption which turned out to be wrong. So we are the product not only of DNA, but also our parents' experience in some fashion, particularly probably with neuropsychiatric issues, things. And that probably explains the missing genetic variation in the genome-wide association studies, if you're familiar with that. So I hope that's not too complicated, but that's the story.
Thank you very much.
My pleasure.
Okay, and we've got one over here. Looks like a microphone.
Thank you.
Thank you.
I just have a question for Professor Mike, because I already asked you this question before, but just want to reiterate it and hopefully give you more details. So, from my understanding, is that because you have emphasized on the enhancer RNA in regulating the genomes, the human gene expression, and the whole unique organisms. So my question is, for RNA to happen, they need to be transcribed from DNA, and that require very complex protein transcriptions of the RNA. So at the point of conception, where did those proteins come from, and how do they get passed on? Like, is it from the parents or from the environment?
Yeah. So the quick answer is that the egg, the ovum, which is a very large cell, and the sperm is just a bullet of DNA, is pre-programmed with all of the essential proteins required to initiate the first round of gene expression in the zygote, or a couple of extra points. It's very important that is asymmetric. So in mammals, the point of sperm entry into the egg creates an asymmetry, so that when the first cell division takes place, you get different on one side and the other, and you can't get proper development without this asymmetry in cell division. Another quick comment I make is to follow Kate's analogy with Lego parts, which I should have mentioned. I mean, a very good way of thinking about the way we are programmed is the proteins are our Lego parts, the enzymes, the structural proteins, etc. And whether those Lego parts are put together to form a house or a castle depends on the complexity of the plants. So Lego parts for animal development are pretty constant all the way from worms up to us. Now there's some differences, new proteins in the brain, etc. But it's the architectural plans that get more and more sophisticated with a relatively constant set of component parts. So I know it wasn't your question, but I think it's a very good and simple way of thinking about, our programming. There's parts and there's architectural plans.
Thank you. Got one here.
Thanks, Ian. Following on from your comments, John. When I went to school and I first learnt chemistry, I thought, oh, this is all Lego with atoms.
Yeah.
And that's what it feels like. But my question is actually for Kate.
Oh, good.
I'm a bit of a simpleton when it comes to things like that. And the more I learn about chemistry, the more it occurs to me that chemistry is just geometry. And that's all it is. What am I missing if I make that assumption?
Oh. Some of it is geometry, absolutely. If I think about a carbon atom, for example, and how it forms bonds, those bonds are in a particular geometry, which is tetrahedral. So, you can't make a structure that doesn't have that tetrahedral shape from a carbon atom. But I would say there's a whole lot more complexity other than just the shape of the molecules. So I could make the same shape molecule from different atoms and it would have quite different properties.
Yeah, but the molecular reactions are all geometrical relationships between the different shapes of the molecules. Certainly in biology, things line up geometrically, and we have lock and key structures. And it occurs to me that everything is just more Lego with atoms.
Absolutely, it's all governed. If you're making a new molecular structure, it's governed by the geometry of the orbitals on the atom. So the directions that those orbitals point is where the bonds form, because that's where the electron density is.
The chemical properties of what you've made are determined by geometry.
Yeah, absolutely.
In the end, it's all geometry.
I think it's more complicated than that, but it's definitely a lot of geometry.
Thanks.
Thank you. Okay, we've got one way over here.
It's really what we in Sydney call a finger on that question, what the Sydney philosophers say. Though it would have been good if we'd had a professor of audio engineering tonight who asked for no feedback. Right? In any case, I'll hold the microphone way down here, I think. To lead on to this question, because you often see these. Is it not? Am I too far away?
No,
yeah, okay. But when we do this kind of modelling, where you had the microtubules and the little robot walking along, you see, the thing that I think when I see that, I think that's really very interesting. But it's like that we understand how a mechanical watch works, and then we just sort of scale down and we make this little robot thing. But surely, when we look at our watch, a mechanical watch, we don't think of fields, we only think of substances. And so we just scale down, but what about the fields, like very tiny electrical fields, because they're ions after all, so they're ionic fields. So you're bringing the substance closer, these little objects are closer to the, and so, which has the priority, but surely there's much more than that kind of Euclidean geometry going on there, and how do the fields affect what we really think about there?
It's on. I'm just going to say yes, absolutely. It's much more complicated than just geometry. I think that was my answer already. But yeah, look, the electric fields, the magnetic fields that, circling electrons make magnetic fields as well. There's a whole pile of other factors that we have to think about. Does that answer your question?
Yeah, it's just that it kind of warps our concepts when we just scale the mechanical bits. Do you know what I mean? And we see this on a screen and we think, oh, okay, at that level, it's just like a watch, but it's smaller.
It's so simple, but it's just tiny. Yeah.
Yeah.
But it absolutely isn't, because if it was that simple, we wouldn't have to spend decades in a lab trying to do it.
Yeah, okay.
And I should hold the microphone further away.
That's right.
That's good.
Somebody else.
Do you want to bring it round?
Oh, you've already had a question.
Pass it
No,
over.
no. Yeah, somebody else.
I'll take the microphone. Let's have a look. So we've got somebody, over here. Would you like to, we used to have someone run around.
A question for Richard, could be a dumb question. You mentioned that chess is incredibly difficult to, what's the word, to put into a computer game, and there was one more that was even more complicated. Is there an equivalent in biology? Take, for example, the chemical molecules or the DNA, RNA. Are people applying computational game theory to these complex chemical or biological reactions? And I'm saying it because one of the first applications of quantum physics, when the quantum computer actually turns up, will be to design new molecules, for example, or to understand the bases of many biological reactions. So does your work translate into that area? And if not, is it possible for it to?
So I guess there are two different things that we can consider here. So one is playing more and more complicated games. And so indeed, I mean, I guess the way in which that has been done more recently, so I mean, I talked about Go and how that had only recently computers surpassed humans in playing Go. And so that was done using convolutional neural networks. And so, I mean, the basic underlying technique is a technique that's been used for very long called Monte Carlo tree search. So that's basically a technique for saying, I don't know the entire game tree, but I'm going to try and find the best strategies I can from this point without knowing all possible outcomes, only seeing a limited distance into the future. But what was new was mating that to convolutional neural networks and using that to train the Monte Carlo tree search. So those techniques have, so convolutional neural networks are now being very widely applied in, I guess, biology and areas like that. The kinds of games I'm studying are very small. I mean, they're as small as the things you saw on that screen there, right? And they don't get very much bigger. They can get, I mean, I'm a mathematician. When I say they're small, I mean they're finite. And when something's finite, the game of chess is small to a mathematician, right? But the games I'm looking at typically are really quite small. And so in that sense, and as I say as well, you're not really studying them to win them. You're studying them to gain a better understanding of fundamentally what's going on when you do a certain kind of computation. So you're kind of trying to uncover some hidden combinatorics or structure behind just the very act of computation, which is not obvious. I mean, you write a computer compiler or something and it just sort of goes off and does stuff. And you're like, great, everything works the way it's supposed to. But you say, what's actually going on behind that? And so that's what we're trying to investigate with this game theory. So I guess it's the same technique could be used sort of towards biology, but the particular applications I have in mind pull away from that, I guess, because of the other desiderata we have.
Thank you, like you're about to add something.
Oh, is that it?
If you could pick up the microphone.
Richard, you gave us 3 possible responses to the question, do you want my number? Yes, no, and here's the number. I'd just like to know what ramifications would occur if you added, I don't know, as a response.
So this is actually quite an important thing to do. So in computation, a very important aspect is just failing to finish. Okay, I mean it's sort of the basis of computation theory is that sometimes your computations don't finish. And in a sense, I don't know. The way a computer would encode that is you just sort of sit there and don't give an answer.
Right.
And so this is really important. And so it's about not playing to the end of the game. At some point, you might just decide to give up. Or you might do something more interesting, which is to sort of continue to loop over and over, asking the same question over and over again and never finishing the game. And so this is a very important aspect of it, and it's to do with the difference between programs that always terminate and programs that only sometimes terminate. And the latter are really important, and they're necessary to get a kind of completely general notion of computation.
So that might make games more interesting.
It does. It makes them much more interesting. Yeah.
Hi, this is a general question for everyone. How's AI impacting your business and in the future, what you believe in?
Do you want to start, Richard?
Okay, yeah. At the moment, so, I mean, okay, there are two things it might impact. One is my research and one is my teaching, and it's definitely having a big impact on my teaching. But for my research, at the moment, the ability of computers to do mathematics is quite limited. So what they can do very well is they can follow techniques and recipes, quite advanced techniques and recipes. So when I talk about my teaching, ChatGPT can do a pretty creditable attempt at undergraduate mathematics assessments. However, they can't go anything beyond that. There's no scope for original thinking. They basically can regurgitate existing formulas, but what you're doing in mathematics is trying to create original thinking, and that's not necessarily possible. Where they are very useful is in computational aspects. So I talked about computation, but in the actual kind of mathematical structures I study, I'm not doing computation. However, there are a lot of people who are doing that kind of thing. So Geordie Williamson at the University of Sydney has a big project to use AI to help solve problems in representation theory, and they are very heavily computational problems. And so there have been recent breakthroughs in kind of reducing the number of operations to perform very basic mathematical computations, like things called taking determinants of matrices and things like this, and multiplying matrices. So these things, which are very, very basic operations, they found more efficient ways to do that using AI. So there are definitely computational aspects for people who are doing computational algebra or something like that. But for the kind of maths I'm doing, we're actually just trying to think up new arguments. It doesn't really impact very much, I think, at the moment.
Yeah, it's a really good question. I think in chemistry, AI has already been used to look at developing new molecular metal organic frameworks. So there's a whole realm of chemistry called metal organic frameworks that looks at building sort of big architectures which are used to store hydrogen gas and try and trap carbon dioxide and all of these sorts of things. And there's a whole project going on at the moment to try and use AI to make better metal organic frameworks, and the predictions are now being tested in lab. So that they're not silly predictions; they look reasonable. There are programs going on looking at, when you do a chemical reaction and you're trying to make a new bond between two molecules, there are lots of factors that you need to optimize. Time, temperature, solvent, rate of stirring. And AI is being used to try and help optimize a lot of those things. And I think on top of that, the machine learning aspects, so not the sort of ChatGPT models, but the machine learning aspects are going to be very powerful in terms of, in my field, predicting what the molecular structure should look like to bind to a sulfate ion. I can make, 85 molecules and one of them will be the right one. But if I program all of those 85 molecules into something that can learn from the structures and the failures, then it's going to be able to predict probably better than I can what I should do to make a better structure. So I think it's going to be very powerful. It's a little bit scary.
I've got a few answers to this, but I'm going to try and keep it to a couple. So using AI, I love it. Makes my life more efficient. Mother of two, play lots of sports, rugby, AFL. I need my time. So I love it. It's great for writing those laborious emails that you don't really want to spend time with. For teaching, it's going to make it hard, but I think collectively in our discipline, we're trying to think of out-of-box ideas on assessment and just trying to embrace it a bit, because I think we're going to need to embrace it at some point. Research. I did mention didn't talk about it much, but we're working in the area of microemulsions for electrolytes with a collaborator, Allegro Energy, and microemulsions, they're based with water, oil, and possibly a surfactant, a co-surfactant, and you mix it in and you get this nice clear solution. So something that should be separated isn't as thermodynamically stable, and you get these micelles. And what we did was we threw this in a battery and we got electron transfer quite readily. So it's a water-based electrolyte. And so now we're like, how many microemulsions can we make? Imagine if we had AI to do that. So we're working with collaborators to produce a model that we can input some data that we've already collected to try and propose some microemulsion ratios that would give us the battery electrolyte properties that we want. So yeah, that's a very long answer.
Thanks. I think it's important to reiterate Kate's point that there's a difference between the popular view of AIs like ChatGPT and machine learning. So I think we're very much using machine learning. So in my case, in dealing with large genomic or RNA data sets, millions, billions of data points. And that's where machine learning is most effective. It needs that number of data points. So we're doing that, and for Richard's benefit, we've just worked out how to use convolutional neural networks, which have basically been developed based on neural processes for analysing image data sets like astronomical data sets, etc. But how to convert non-image data, such as we deal with, into quasi-image data so CNNs can work, and that paper's just been accepted. And we're using it to actually try to parse the structures in RNAs that bind particular proteins so we can put some sense into how these molecules work. My quick second comment is that my conviction is that the future of biomedical research, perhaps all biological research, is going to be not the reductionist past, sort of looking at one protein or gene or, surface molecule, which is important to get down and dirty, but rather huge data sets. You imagine you've got millions of genomes in medical research, millions of clinical records, pharmacological records, and most importantly, smart sensor data. And I'm an advisor for a company in Britain that's got 1 million Brits signed up with all of their smart sensor data as well as all the other things downloaded from Apple by agreement. And this is going to allow us to find patterns in very complex sets of data and track down the genetic and biochemical physiological basis of complex disorders like neuropsychiatric disorders, arthritis, etc. So I'm really pushing the federal government to establish data centres in Canberra that are privacy protected, etc., to allow this ecology to evolve. Because I think that the new generation of discoveries is going to be machine learning on these huge data sets that we have to accumulate and then make available for research and ultimately for health translation.
And the microphone's over here somewhere. No?
Hello?
This gentleman.
Is it on? Can you hear me?
We can hear you.
Okay. I have two questions, one for Professor John and another one for Professor Katrina. The first question is like...
No, we can't hear you. I'm wrong. Put the switch up. There's a switch on the side, a little slide that goes up. Not on the black box, on the cylinder of the microphone.
Right. No?
It takes a second.
That's it.
Okay.
There's a delay.
Yeah, two questions. The first question is for Professor John. Now, the brain structure and the intelligence is completely defined by RNA, or we can go beyond. For example, now physics look into particle physics. We have a standard model of particle physics. Now, do we stop from RNA because we are studying biology, or can we further going into the more fundamental level and we can understand the intelligence and the brain structure? That's one question. The second question is for Professor Katrina. I have read about...
No, could you stop a moment?
Okay.
Can you let them answer first, because no one will remember.
Okay, good.
Right. So the short answer is no. Particle physics won't help us understand cognitive processes.
And as I quipping to, I think, Kate earlier, physics won't even explain chemistry. Once you get past 2 or 3 atoms, you just can't do it. So that's not possible. The data that's coming out is that in nearly all the transactions in the brain, including synaptic events, are all RNA mediated. The proteins are the effectors, but the RNAs tell them where to go and when to do it. So it's early days, but there's a terrific group at the University of Queensland doing this and showing a lot of they've just looked through a new door and showing all of these RNA transactions happening at synapses during learning. So I think about biology in terms of information more than chemistry. It's transduced by chemistry, but it's the RNA that contains the information, and the proteins are, excuse me, biochemists, the dumb effectors of what the RNAs tell them to do. But physics is not going to help us understand the brain.
Okay, so we'll pass the microphone on. And your second question?
Yeah, the second question is, you present something about microtubules. I read a paper about explaining orchestrated collapse of microtubules in the brain has a quantum effect. So now, is Australia focusing on biological inspired quantum mechanics? Is there something in Australia looking into?
I don't think I can answer that question. So, look, there is a lot of quantum physics happening in Australia, and it's got a lot of different applications. I think, as Susan said earlier, one of the first things that's going to happen is that quantum computing will be applied to chemistry, solving chemical problems. But I don't know the answer to the biomechanics question.
Oh, hold on. John's got a question.
For James. For Richard, sorry. Richard, what you were talking about reminds me very much of a mathematical domain pioneered by Mike Fellows, who used to be at the University of Newcastle, called parameterized complexity, where he said if the system is too complex, too many variables, the search space is too large. And so the only way to search it is to try to work out the subspace, which is going to, maybe that's what you're talking about with Go. Does your approach intersect with parameterized complexity in complex systems, whether it's in biology or anywhere else?
Let me think. My inclination is to say no. So again, the focus is, so there are games, and there are complicated games, and then there are games which describe the semantics of kind of just the very fundamental structures of computation. And those are very simple games, and despite their simplicity, they're very complicated, as you saw. I took a game which is just 3 layers deep, and just getting some understanding of what all those things are, a real understanding of what those things are, is very, very hard. And so somehow, so the motivation behind this is that before the games, there was still this complicated structure where you could keep asking question upon question upon question up to sort of as many layers deep as you wanted, was there, but people didn't really know how to handle it. And so really what you're trying to do is it's less about a game which comes from kind of some observed real-world system than a game which comes from trying to understand what exactly computation is at some sort of fundamental level. And I guess because of this, you kind of don't really care about complexity, because the complexity, if you like, is all in the implementation. And you're not really caring about the implementation, you just want to have a mathematical representation of what this computation is.
Fantastic. Now, we've got someone here. Yes.
Hi, this is the final question, but actually it has got A and B questions. Professor Katrina, so you were talking about this protein and these anions you were working on. I'm currently teaching WEA one course called Life in the Universe. So I'm an astrophysicist, but I'm trying to teach about the origin of the life, on Earth and so on. So I got a very little idea about the cell formation, which has got RNA, which can be replicated with DNA, and then it has got protein, and it has got the lipid, which is the membrane. So I know that you're talking about the protein and anion, but I also saw some of the form in one of the slides, which looks like lipid to me, because they have these two legs and one ball. They can be hydrophilic or hydrophobic. Depending on that, they will be arranging themselves so that they can form that clear membrane around the new RNA and protein. Have you done anything with anion thing with the lipid? Like, is there any connection with that? This is my first question. Then the second question I'll ask right away. You have been showing all these genetical codes and things like, this architecture. It reminds me of, can we also build some X-Men or Hulk or Spider-Man at some point with this? Thank you.
Yeah, okay, so the first question was about the lipids, and yes, absolutely, we're looking at those lipid head groups. The cell pictures that I showed at the end of the talk were actually molecules that are binding to the membrane, each lipid molecule in the membrane. So yeah, you can look at molecular recognition of all different types of things. They don't have to be anions, they can be cations, they can be neutral molecules as well. The same principles apply about finding something that matches the size, the shape, the charge of the species that you're interested in binding to. And your second question, look, I have a publication that I use in teaching that shows molecular hats on different stick figures. So there's an actual publication in the Journal of Organic Chemistry that looks at changing the hats by changing the molecular shape. So yes, absolutely, we can think about building all sorts of things.
Thank you.
Thank you very much.
I've been asked to actually, whoop.
Do you want this one?
I guess I know how to press the button. Yes, no, yes. That's the way. Okay, well, I've been asked to wrap it up. This it was absolutely fantastic. Great questions, great talks, fantastic. I can't resist. I have to ask you something very simple. Would you like to live forever? You 5. You as well, yeah? Would you like to live forever?
Absolutely.
Yes.
Only if I can stay young.
That's a middle question, middle answer. Yeah. How about you?
Nah.
No. So we have a 2-1.
Yeah, I'm a no as well.
2-2.
I'd love to see what science is going to do over the next 2 centuries. I'd just die for that, if you use the pun. However, I'm with Kate, so my idea is I want to download what's in my brain. And I don't have to clone myself, just put myself in a new body, because you don't want to feel like you do it.
The good news for you is only 20% of humanity would like to live forever. But when you look, who is that 20%? 90% of these 20% is actually older than 50. So that means that you are young. Okay, so that's good for you. Let me thank our sponsors tonight, Laboratories Credit Union. Absolutely fantastic. Thank you, guys. Fantastic. Of course, our speakers and everyone, and my colleague from chemistry, Raki. I have something also, to say here, right?
Thank you, Miroslav. It's been a really fun night, so I hope you've all enjoyed it. I'd like to thank our wonderful speakers. You've all contributed your own little insights into this thing of science. And I guess one of the things that I wrote down was actually from John's talk, which was, we had this understanding that genes were composed of bits that were making proteins and all the rest was junk. And it is not our purpose as scientists to work out what the purpose is of the junk, if you like, and understand, nature has a wonderful way of making purpose for everything So, I guess we all do try to understand that in our own special way. Anyway, so thank you to each of you. It's been wonderful. I have the honour of giving some little gifts to our speakers. The first one is for John. Thank you so much.
Thank you very much, everyone. I had fun, and I hope you did. Thank you.
Next one for Renee. A wonderful communicator, I have to say.
Thank you.
Loved your presentation. Well, my friend Kate. Thank you so much, Kate. For Richard, do you do stand-up comedy as well? Sorry? Do you do stand-up comedy as well? It was terrific, kept me entertained. It reminded me of the days in a computer lab at university and why I became very good at English syntax after all the syntax errors I was getting with my computer programs. And finally to Ian for convening this wonderful Q&A session. Thank you. Thank you. Over to Fred.
I'll just make it quick and just want to thank everyone for coming out tonight. It was a real enjoyment. Another fantastic Frontiers of Science Forum. It's the fifth one. Looking forward to the sixth year next year. But thank you. God bless. Take care and hopefully see you in the near future. Thank you.